Hello. For this session, our speaker are Ryan already. I welcome one speaker, we have Malik. Our first speaker is Dr. Session Sades, with Malik, Canada. Our second speaker is Dr. David Windes, he is working as a senior scientist, officer at Global and Development Research and Development Agency, Geneva, Switzerland, established a PhD candidate to address the development and treatment of drug resistant bacterial infection. And our first speaker is Dr. Balram Muslim, working as an scientist at National Chemical Laboratory. I hand over this session to Sharon.
Hello. Thank you. Good afternoon to one and all present here. At the outset, I am very much thankful to organizing committee for giving me the honor as the chairperson for this particular session. Honorable Dr. Gazbie sir, Professor Aswar sir, galaxy of intellectuals, all faculty members, all participants, students, and other delegates. So, you have heard very good lectures before lunch from the Dr. Dikshit. And again, the first lecture of this session is also very, very interesting. And for this, we have Dr. Shailesh Subhasrao Sabre, the scientist working at University of Alberta, Canada. In his past experience, he has worked as a senior research scientist at Graduate Institute of Environmental Engineering, National Taiwan University, Taipan. Also, the project research scientist, Chemical Engineering Department, University, Ravira, Spain. Project assistant at National Chemical Laboratory, Pune. And it's a honor and that is for Sant Garge Baba Amravati University, as he is the alumni of the Sant Garge Baba Amravati University. He did his MSc chemistry from the Sant Garge Baba Amravati University and did his PhD in chemical sciences and engineering. He has published number of research papers in reputed journals and also presented papers in conferences. His area of research is catalysis in environmental and green processes, development of heterogeneous catalysis for innovative water and wastewater technology, water treatment, science and engineering, and the hazardous pollutants. So, without taking much time, may I request to Honorable Dr. Sabre sir, please go through your topic.
पोस्ट का शेअर करेल ते त्यांना कॉल करून सांगा थोडं कॉल लागेल का नाही तर मेसेज चॅट बॉक्स मध्ये पोस्ट का शेअर करेल
Please let him allow to share his screen from your side.
Very good afternoon, everyone, and thanks for the nice introduction. Myself, Shailesh Sabare. Today, I'm going to deliver my talk, which is based on performance and mechanism in catalytic oxidation processes for the degradation and mineralization of emerging contaminants from water and wastewater. Before that, I would like to share my short research journey with you guys. I am born in small Vijayanakwari, which is near to the Kunjamuzri in Amravati district. I had completed my schooling from the government school. After that, I have completed my graduation in science and post-graduation in chemistry from Shivaji Science College, Amravati. Hello? Okay. Hello, can you hear me?
I can hear you, Shailesh.
Okay, thank you. Hello. Maximize slide. Maximize slide. Maximize slide show शायद. ओके ठीक है. ओके ठीक है कर लो.
हेलो
Hello.
हेलो. हाँ शैलेश सर. फुल फुल स्क्रीन मोड घ्या सर.
Hello.
हो सर फुल स्क्रीन मोड घ्या.
And can you see now the full screen? Hello.
पीपीटी जी आहे ना ती फुल स्क्रीन घ्या.
हा, PPT full screen दिसत आहे का आता? हॅलो.
हा स्क्रीनवर पीपीटी दिसत आहे सर फक्त.
हा, स्लाइड शो दिसत आहे काय?
हा हा ओके.
दिसत आहे स्लाइड शो?
हो.
केलं ना full screen चं? कशा म्हणतात?
सर कंटिन्यू प्लीज.
Good afternoon, everyone. I am extremely sorry for the inconvenience. So today I am going to discuss my topic, which is based on performance and mechanism of catalytic oxidation processes for the degradation and mineralization of emerging contaminant from water and wastewater. Before that, I would like to share my short research journey, you guys. I have completed my master in chemistry from Sri Shivaji Science College, Amravati, and then I had been working for 6 months in Tata Western Company. After that, I got opportunity to work as a project assistant at National Chemical Laboratory, Pune, where I worked for 2 years. And from there, I was looking for the PhD fellowship in abroad, especially Europe. Then I got PhD fellowship from Ministry of Science and Innovation from Spain, where I have completed my PhD. And during my PhD, also I got the mobility grant from Germany, where I did my research stay at UFZ Leipzig. After that, I had been working as a project research scientist in the same university, University of Rovira Virgili, with Professor Francis Medina. After that, I got Ministry of Science and Technology Fellowship from Taiwan, where I worked 40 years. And after that, I moved to the Canada, University of Alberta, in Edmonton, where I was working as a senior researcher. So I'm going to the topic. As we know, water is life, treated right, and we are living in the world of increasing water scarcity because the availability of fresh water is essential for the development of any nation, disease-free society, and quality of life. But the global neglect of water and waste management has serious consequences for human health and environment because numerous human activity, industrialization, and population growth increase the wastewater and waste material in the environment, which has threatening effect for the human life and aquatic life as well. Because of that, as per UN Environmental Program report, in coming 10, 20 years, the budget for the management of water technologies and management is going to increase by 25 times. Thus, we need the specific solution and protocol from the government organization as well as from the private player. In this work, especially, I had studied emerging contaminants and phenolic contaminants, which is introduced in the water bodies through the pharmaceutical and personal care product every day. The emerging contaminants are the contaminants which are unknown, often in very small amount of concentration in some compounds, often in the ppb level, which are not regulated, but it shows the environmental and human health effect, which are the unknown. As per European Union directory, they have created the watch list for the several pharmaceutical and personal care products. Even perfluorooctanic and perfluorosulfonic compound, these are the hot topic nowadays. In this work, I have studied the chlorphenic acid, which is the primary metabolite of lipid regulator drug, which shows several harmful effects on human health and aquatic system. From the phenolic compound, I have selected the phenol, which is toxic and hazardous, often from the several industries like paper and pulp, textile, pharmaceutical, rubber, gas, and coke, which has threatening effect as protein degeneration, tissue erosion, paralysis of central nervous system. It can damage kidney, liver, and pancreas. We have the conventional technologies to treat this type of recalcitrant compound, like the sedimentation, flotation, filtration, absorption, coagulation, and biological treatment process. But the complete removal using this technology is not possible. Even the most harmful byproducts are forming and remain in the water bodies, groundwater, surface water, as well as in drinking water. Thus, we need the implementation of environmental process beyond this traditional process to remove these contaminants completely from the water bodies. Therefore, we introduce advanced oxidation processes, which is the promising technology for the water remediation, which is based on hydroxyl radical. This is the environmental friendly process as we can apply it at ambient temperature and pressure. Heterogeneous catalysis along with advanced oxidation processes could be the one of the alternative to treat this type of compound on the basis of secondary or tertiary treatment processes. As on the catalyst surface, adsorption, reaction, and desorption, this type of reaction occur. Heterogeneous catalyst is the material which we can reuse after the reaction. The best heterogeneous catalyst material has several properties that it is easy to separation. We can recycle it and reuse. Generally, this type of catalysts are reusable and eco-friendly. Here, mainly, I have studied catalytic ozonation, where ozone as oxidant use, and penton-like process, where copper and iron type of catalyst use. for the degradation of chlorofluoric acid and phenolic compound. In Fenton-like process, we have used commercial hydrogen peroxide as well as hydrogen peroxide was generated in situ using formic acid and oxygen. For this purpose, several families of catalysts were synthesized like hydrotalcide and spinel type catalysts, iron oxyhydroxide derived catalyst, copper-based catalyst, and zeolite-based catalyst for the adsorption and oxidation process. Hydrotalcides are double layer compound which has high surface area, basicity. These materials are thermostable in low crystal size and it has a memory effect. That's what we have selected this material. Therefore, different type of hydrotalcides using magnesium and copper aluminum oxide, magnesium iron aluminum hydroxides were synthesized and calcined at different temperature from 400 degrees Celsius to 900 degrees Celsius. All these materials are characterized using different characterization technique like XRD and BET surface area. So from this result, copper aluminum oxide spinel phase and corundum crystal phase and tenorite crystal phases were obtained. After that, we have used this catalyst in degradation of chlorofluoric acid for the 2-hour and 6-hour ozonation process. From the result, we can see complete removal of chlorofluoric acid means complete conversion of chlorofluoric acid was obtained within 15 minutes of reaction. But oxidized byproducts were still in the water bodies. From the result, we can see as compared to the single ozonation, more than 40 to 50% of effect clearly observed using the catalyst. When we calcinate catalyst for the higher temperature, the spinel phases form, which shows the better activity and stability in the process, achieving more than 80% of mineralization. So among all these catalysts, magnesium iron alumina and copper magnesium aluminum oxide catalysts are the most stable and active catalyst, which is giving complete CFA removal without any leaching of iron, and it gives the complete degradation and mineralization of the chlorofluoric acid. And this work has been published in Applied Catalyst Environment. Second family of catalysts we had studied iron oxide derived catalyst, which is abundant in nature. It can easily synthesize in lab using different synthetic protocol. Also, we can operate this type of material in wide operating pH range, and it has controllable iron leaching properties. Therefore, commercial iron oxyhydroxide were bought and calcined at different temperature. Also, small amount of palladium were impregnated by impregnation method, and iron oxyhydroxide impregnate of alumina and zirconia are also synthesized and calcined. These are the physico-absorption property of this catalyst along with the XRD results. From here we can see mainly maghemite and hematite was the phases obtained after the calcination. Similarly, like a hydrotalcide compound, complete removal of chlorofluoric acid was obtained in 15 minutes of reaction. Among all these catalysts calcine at 250 degrees Celsius and palladium impregnated catalyst shows the better activity, achieving 80% of removal within 2 hours of ozonation. Similarly, we have used this catalyst in ozonation process as well as in Fenton-like oxidation, where hydrogen peroxide was used in the commercially. So from the result, clear catalytic activity has been seen using palladium iron oxide hydroxide catalyst calcined at 200 degrees Celsius, achieving 100% CFA removal in 2 hours of reaction time in Fenton-like process. Even though higher TOC removal degree of mineralization achieve more than 60% within 30 to 60 minutes of reaction time. In this work, we had also studied the oxidation degradative products intermediate mechanism which is not shown here, and this work has been published in Catalysis today. The next family of catalysts we had studied is copper-based material. So for that, we have developed copper double catalyst, copper hydroxide and spinel catalyst, and copper aluminum oxide catalyst by co-precipitation method and calcinide at different temperature level. So among all these catalysts, we can see the copper alumina catalyst shows the better result, achieving more than 81% of TOC removal without any leaching of copper. When compared to the copper double and copper hydroxide, it shows 20 to 25% more TOC removal. Other catalysts like copper double and hydroxide material shows the copper leaching. So homogeneous contribution of this copper leaching was also studied. Therefore, copper used in the reaction and tested in the catalytic oxidation process, which shows around 38% of TOC removal. Obviously, this catalyst, copper aluminum oxide catalyst, is most stable and active catalyst. Therefore, we perform thoroughly catalytic characterization of this catalyst. And from the XRD study, copper oxide and copper aluminum oxide spinel phase were obtained. And from the XP study, copper 2 positive is the oxidation state observed, and high acidity using ammonia TPD. Lewis and phosphate acid sites were studied using Pyridine IR study. There we try to understand the mechanism in catalytic ozonation process. As catalytic ozonation process in aquatic media, because the system is very complex. So we tried to understand what is the possible steps occurring during the reaction. So these are the 3 to 4 probable ozonation mechanism process where the ozone as oxidant can directly oxidize the organic molecule to form the oxidized product. Other possibility is that where ozone can be adsorbed on the catalyst surface to form the hydroxyl radical, which can attack to the organic molecule to give the. Another possibility is that the adsorption of organic molecule on catalyst surface and direct ozonation occurred on the catalyst to form the oxidized product. And the next possibility is that the both ozone and organic molecule adsorption on catalyst surface to give the oxidized product. To make it more clear and to understand the mechanism, in situ ATI-IR technique was implemented to know the surface changes occur during this process. For that purpose, ATI-IR spectroscopy was used, which is very easy to use and fast versatile techniques, which can do the identification of active site of the catalyst. Also, we can employ this technique for the adsorb and dissolve species at solid liquid interface. So to perform this type of experiments, aqueous ozone was passed through the Zn selenide ATR cell, where catalyst bed was prepared, and then spectra was measured in the continuous flow of ozone and without ozone to know what is happening. So these are some experiments we had performed in the presence of water and deuterated water. From the figure A, we can see when there is unozonated copper alumina catalyst on the bed and the spectra was measured, 1648 centimeter inverse, there is a spectrum occur. So that is spectrum for catalyst, but in the presence of ozone, there is 136100 new spectrum occurred. This is for the active site of catalyst, where chemi-absorbed water react with ozone to form the active species. And this is the reference spectra of chlorphenic acid obtained at 1500 degrees. So this surface active species has the stronger interaction on catalyst surface. as we had seen from the pyridinide study, the acidic side of the catalyst, which form these active species and further react with the catalyst to form the hydroxyl radical, which can participate in the reaction for the degradation of the organic compound. To make it more clear, we had performed the experiment in the presence of phosphate, as phosphate is an inhibitor which can inhibit the formation of hydroxyl radicals on surface. And here we can see in the catalytic ozonation of chlorhydric acid reaction, when we use the phosphate buffer, there is clear degradation difference when we compare to the single ozonation. Similarly, in situ ATR-IR experiments were performed using the phosphate and water, where we can see from the figure the peak which is for the active species, which is appear at 1361 centimeter in mass, was disappeared in the presence of phosphate. It means that the reaction takes place on the surface of catalyst, where this chemi-absorbed water react with ozone to form this type of species. Also, the degree of mineralization in the presence of this catalyst was studied at different chlorhydric acid concentration and different initial pH, where the efficiency of this catalyst also been demonstrated, and this work has been published in Applied Catalyst Beyond Moment. Next family of catalyst we have studied is zeolite-based material, which has very good adsorption affinity. This is highly porous material. It has also shape selective property, and this is the environment friendly material. Therefore, to implementation of adsorption and oxidation concept, we have choose this material, hydrophilic FeZSM5 catalyst and hydrophobic FeZSM5 catalyst. Also, small amount of palladium was impregnated by impregnation method and followed by reduction by NaBH4. These are the characteristic properties of zeolite, where we can see the BT surface area and silica alumina ratio. XRF analysis shows the palladium content in the respective catalyst. Adsorption isotherm was also studied for the phenol. And from the results, 35% of phenol adsorption has been obtained on hydrophobic zeolite, whereas 10 to 15% of phenol adsorption was obtained on hydrophilic zeolite. Then we have introduced this catalyst in penton-like oxidation process. And these are the results, where among all these catalysts, the small amount of palladium impregnated catalyst, that is 0.1% palladium on FeZSM5, shows the better results. Therefore, we had selected this catalyst for the recycle and reuse purpose, and it demonstrates a good activity, where we can see after 3 cycles, it shows the activity decrease around 20%. Also, efficient hydrogen decomposition was observed. Similarly, hydrophobic palladium FeZSM5 catalysts were implemented in phenol degradation reaction and H2O2 decomposition. And 0.1% palladium hydrophobic FeZSM5 shows the better activity, achieving total phenol conversion within 4 hours of reaction, and better decomposition of hydrogen peroxide was obtained. So, we demobilize catalyst shows the great utilization of hydrogen peroxide and total conversion of phenol in 4 hours of reaction. So these are the results where we had used the commercial hydrogen peroxide. Similarly, in situ hydrogen peroxide was generated using formic acid and oxygen, where a decomposition of formic acid occurred to form carbon dioxide and hydrogen, which reacted to oxygen to form hydrogen peroxide. And similarly, catalytic decomposition of hydrogen peroxide result into the formation of hydroxyl radical, which can attack and oxidize the target organic compound for the purpose of total degradation and mineralization. These are the results for the in situ generated hydrogen peroxide reaction, where 0.5% palladium impregnated on APZ-5 catalyst shows the better activity, around 65% of phenol conversion, and 58% of formic acid decomposition. So from this result, we can conclude that stronger adsorption enrichment of phenol has been shown using this type of materials, which are the environmental friendly materials, and we can perform this reaction at ambient temperature and pressure. So combined approach of adsorption and oxidation has been achieved for the better catalytic system, and that's what we can introduce this catalyst in for the industrial purpose also. These are the few research scholarship and opportunities are available in the different part of world. The enthusiastic young researcher who want to pursue their career in abroad, these are the some link you can have a look. Different government fellowships are available in Quebec, Ontario, and every year there are number of fellowship in University of Alberta for the master study and PhD study as well. These are the links for the research job and master and PhD study in North America. Similarly, in Europe, there are number of organizations offering different type of fellowship, scholarship award for the master and PhD student. You can have a look in ETH Zurich. Also, the prestigious fellowship like Humboldt Fellowship, GSPS Fellowship in Japan, Singapore, NSC Fellowship. Also, you can have the Ministry of Science and Technology Fellowship in Taiwan. And this is the link for the numbers of fellowship and awards. You can have a look into that. Finally, I would like to acknowledge University of Virar Arjali for my PhD study, research and funding. University of National Taiwan University for the researcher position and funding. Also University of Alberta. Thank you for your kind attention. Finally, I will really appreciate my thanks to the organization committee for giving me this opportunity. Thank you.
Thank you, sir. चला. Hello. Oh. Hello. On the synthesis of the catalyst and its application, particularly the remove in situ generation of the H2O2 in Fenton-like processes, and catalyst. So if anybody has any query regarding this topic, you can put your question in chat box. So that, yes. You can put your this lecture. Thank you, sir. So next, our next speaker is Dr. Ravindra Zumre, the Senior Scientific Officer at Global Antibiotic Research and Development Partnership, Geneva, Switzerland. He has been awarded with the PhD at University of Pisa, group of Professor under the guidance of group of Professor D. Pini and Dr. A. Mandoli, Pisa, Italy. His past experience include that is science writer at Faridabad University and Helmholtz Institute for Pharmaceutical Research, Germany. Project leader and Marie Curie Action researcher at Helmholtz Institute. At Germany. And also the senior postdoctoral researcher at the University of Groningen group of Professor S.R. Horethan, Groningen, Netherlands. Also did his postdoctoral researcher at the Institute of Science and Technology group at Italy. He is recipient of many research grants and awards. His teaching experience is includes that is the HRPS Germany, co-supervised 1 postdoc and 3 PhD, master's and bachelor thesis project also under his able guidance. He has published a number of research papers in the and beside that, I request Dr. Ravindra Zumre sir to please and before starting his topic, he will also introduce about his research area. So without taking much time, again I request Dr. Zumre sir, please go through your topic, sir.
Thank you. I hope you can see my screen in full now. Okay. Thank you very much, ma'am, for your kind introduction. And I'm really thankful for the organization committee of National Workshop on Modern Approaches in Material Research for giving me this opportunity to talk about my research. So today I will talk about role of chirality and drug discovery. I hope everyone can hear me properly now. Please shout out if not. So Madam already told about my journey. So I will start with my talk with my short personal journey. Then I will delve into drug discovery process and timeline, give you a brief overview how the drug discovery process works, mainly for the students. And then I will told something about my current organization where I'm working, which is GARDP, how we work and how we develop new medicines and how we'll access it to the world. Then I will go more into the science. So talk about role of chirality in drug discovery and challenges, giving you one example where we develop a catalytic enantioselective process to develop this chiral compound, followed by outlook. And then, as Shailesh already mentioned, I will also give you a brief overview how the research opportunities are there for the young and enthusiastic researchers. So talking about my personal journey, I did my BSc from Jagdamba Mahavidyalaya, Cholpur. Then I did my MSc from Sri Shivaji Science College, Amravati, like Shailesh did. And after that, I also joined project assistant at National Chemical Laboratory in Pune. So similar path as Shailesh. After that, I got a Galileo Galilei PhD fellowship in Italy at University of Pisa, where I worked on developing organocatalytic chiral organocatalytic reactions. Followed by, I went to Netherlands in University of Groningen, where I did my first postdoc, and there I developed catalytic processes, enantioselective catalytic processes, new reactions. And I will talk about that in my talk in a few minutes. After that, I wanted to apply this kind of methodology for some application, and there I wrote a grant application for Marie Curie grant, and where I used this kind of methodology for drug discovery purposes. After that, last year, I joined a global antibiotic research and development partnership, which was started by WHO as a senior scientific officer in Switzerland, but working from UK at the moment. So let's start with the drug discovery process and timeline. So I will try to give you a really general overview of a drug discovery, so all the students at least know how the drug discovery process works and how much time it requires in total. So usually the drug discovery process starts with selecting biological target. What is biological target? So just take an example of coronavirus. You know that for coronavirus, it tends to replicate, right? And it has also spike proteins which attach to the human cells, right? So for this replication, there are enzymes which are required to catalyze this process. And similarly, for the spike proteins, there are also other enzymes, which we call them a target in a drug discovery process. So whenever I will talk about a target or enzyme, just keep in mind, these are kind of a function of bacteria or virus, which are essential for their growth or keeping them alive, okay? So the drug discovery process starts with either identifying these kind of targets or a whole bacteria or virus. And how do we do that? So there are different science involved in that. So bioinformatics, genomics, and genetic target identification. So this kind of methodology which we use to identify these kind of targets and viruses and bacteria. So once you have that information, so next is to test a lot of chemicals. So what I mean by a lot of chemicals, this process is called hit compound generation. So what is hit compound generation? So there is like, we want to find a chemical which can stop this process in a bacteria or virus to grow. And there are different ways to do that. One is called high throughput screening. What is high throughput screening? It is to screen thousands and thousands of compounds and to check which compound can stop inhibition or stop or inhibit this growth of bacteria or virus. So this can be done either on the target, or that means the enzyme, or a whole bacteria or virus. And the process, when we test it on whole bacteria or virus, is called phenotypic screening. And when we test it on a particular part of that target, which is called a particular part of that organism, which is called target or enzyme, that's called target-based high throughput screening. And recently, there are also computational screening going on. And that's a hot topic at the moment, where you don't need to physically test each compound. But what you can do is with the computational screening, you just screen these compound structures on a computer and check which compounds bind really good in these enzymes, and then carry it forward. So this is how we find the hit compound, right? And that involves some initial synthesis, what you do in the lab on day-to-day basis. So then the next step is to find a lead compound. What is lead compound? Difference between hit and lead compound. Lead compounds are the compounds which are advanced, which shows higher affinity towards the target, or which can kill bacteria or viruses more effectively than the hit compound. So how we go from hit to lead compound is to try to do some modification on the compound. Just take an example. You have a compound with a methyl ring on the benzene. So we see that this compound is killing bacteria, but not 100%. Now we try to change that methyl group maybe with some halogens and see if that works good. Sometimes it works, sometimes not. But if it works good, that means that replacing methyl with a halogen is good. So that's how we kind of optimize these compounds forward. So there are a lot of synthesis process going on in between. So we synthesize, we test the compound, we check which one is better. So once we get this lead compound, which has better activity than the hit compound, and what the process of checking what modification on compound reflects to its activity is called structure-activity relationship. You can read about it, and it's really fascinating how it works. And this process requires really advanced chemical synthesis. So multi-step reactions going on there, and there's back and forth of synthesis and testing going on. The next one stage is to select the drug candidate. So out of these lead compounds, we try to get a drug candidate which is even better, right? So we don't want to test thousands of compounds at this stage, but there are like maybe some hundreds of compounds. And what we do, instead of checking directly into the humans or cells or animals, what we do is to do a cellular disease model. What is cellular disease model? So it's like, just take an example of coronavirus, right? It first affects your lungs. So what usually we do is to take lung cells, grow them in a petri dish, and incubate them with these compounds, right? So first infect that cells with coronavirus. So coronavirus goes into the lung cells, but that's not into the body, that's in a petri dish. And then what we do is to add some compounds on the top of that and check if this coronavirus is now dead. So this is called cellular disease model. So you don't need to go into the animal yet. And there we also check the selectivity. So is it selectively killing the virus or bacteria, or it's also killing the cell? If it is killing the cell, that means it will also kill the cells in your lungs. So that's not good. So what we need is a compound which can selectively kill the bacteria or virus. So this study does like maybe it's called drug candidate profiling. And after that, when we see that, okay, we found a compound or two compounds maybe, which are perfect, they are killing the bacteria, not killing the human cells or the animal cells, then we go to the preclinical development. These are like animal models, you know? So just take an example that we take a mouse, we infect him with some bacteria or virus. So for instance, in the lungs, so respiratory tract infection, right? And then we treat this mouse with this compound, with this chemical compound, either orally or intravenous in the veins, right? And check if this infection is now, after few hours or few days, if this infection is gone. So this is called preclinical development. There are a lot of things going on in this preclinical development, and this can go for years and years. So we can also check their pharmacokinetics, pharmacodynamic study. We also check if this compound is toxic to the animal. So if we're giving this compound to the animal, if the animal dies, then yeah, there is something wrong with this compound. So all this information we gather and we feed back again, we go to the lab, we change some molecule, we again test them into the cellular disease model and then the animal. And this process goes on until we find a final compound, which is really great, which kills the bacteria or the virus, and it is safe for the animal. And only then we go to the human clinical trials. And there are different phases in there, phase 1, phase 2, phase 3. That's called human clinical trials. If you want to read about it, go read into the Google. You can easily find what clinical trials are. So what we do in this case is mainly safety to the human. So we check if there are some side effects. And if there are some side effects, are they lethal? Means, are they killing the host? I hope not, because that's what we don't want. and how good is tolerable, how easier to get this medicine. Preferred medicine is by oral route instead of the injections. So that's how we try to develop the drugs. But sometimes the drugs is not absorbed well in the body when you take it in orally. So then we develop a drug which we can also inject in your veins. So once everything is, these clinical trials are done, then the drug comes into the market. So before the market, it needs to be get the government approval. And in different countries, different organizations work for that to check if everything went well from the human clinical trials. And then it is introduced into the market, but it's not end of the story. After even in the market, there is a post-market survey. So what we do usually here is to check if you take this drug for long term, if there are some side effects. And many times it happens that the drug is withdrawn from the market because it shows really significant side effects which were not present when in case of clinical trials. So the whole process of this drug discovery, which lasts for years and years, can and you spend billions of dollars on that, and then still it can be taken out of the market. So just to give you an overview how much work it needs. So for the drug discovery process, let me go to Lisa. Yeah. So we start with, this is an average. Sometimes it goes to tens of thousands of compounds. So we start with tens of thousands of compound in a drug discovery stage, which is this hit compound generation. Then we go to the preclinical. So this thousands of compounds goes to around 200-ish compound in a preclinicals. And then when it reaches to the clinical trials, it goes to 4 or 5 compounds, sometimes even 1 compound. And at the end of this story, which gets only 1 drug. So a whole process requires at least 10 years. There are some examples where the drug is from the discovery till the market reach in like 5 or 6 years, but that's rare. So it requires a lot of research and a lot of clinical trials, long years of that, and then the government approval. So this is how the drug discovery process works, and that's why you find that there are no new drugs coming in the market like on a regular basis. Okay, so my current organization where I work is a global antibiotic research and development partnership, which was created by World Health Organization and DNDi, which is Drug for Neglected Diseases, other organization which was created by WHO, and it was started on 2018. So just 5 years before we started this organization. So what is our mission? Our mission is to accelerate the development of these drugs and give the access of this drug to general public and mainly for the lower and middle income countries. So we work together with public, private, and nonprofit partners, and we try to preserve the power of antibiotics for the generations to come, because you know that the antibiotics nowadays are not working. For the serious infection you see in the ICU patients, the antibiotics is not working because the bacteria is getting used to that. And this is all because of heavy use of antibiotics. When you get like cough or cold, you don't need antibiotic at that time. But people tends to get antibiotic from the pharmacy, from the medical, and this increase this problem that the bacteria get used to this kind of antibiotics. And then when you really need the antibiotic, the best antibiotic present in the world is not working. So we want to prevent that, and that's why we are in this field. So where do we work in this whole drug discovery process? So we work in the early phase, and this is the Department of Discovery and Exploratory Research where I work. And here we work from hit compound generation till the drug candidate. And other departments in GARDP works also for, we fund all the clinical trials, we fund all the production of these compounds as a drug. And like, we are not a company, we are not an industry, so we don't want profit from it. So we are non-for-profit organization. So what we also do is to give access to this new medicine to lower and middle income countries in Asia, Africa, and South America free of cost. So we get funding from different government and sometimes also the private partners. And we try to get this drug at least minimum cost to the general public. So this is for preclinical, as you might wonder that why we are not working in preclinical development. We outsource it. We outsource it to different organizations, and there are other organizations, non-for-profit organizations, working on that. So we don't go into, or we don't try to replicate what others are doing. So just to give you a brief overview what I do. So I work on serious bacterial infection and sepsis. So our research area spans in 5 different projects. So which starts with small molecules. So we test small molecules. We also work on natural products. We work on potentiator. What is potentiator? Is to find a compound which can enhance the activity of present antibiotics, right? You administer this compound in combination with the antibiotic, so it can enhance its activity. So it's not an antibiotic, but it just try to enhance the activity of known antibiotic. Then unrealized target. As I mentioned, what are the targets? These are kind of enzymes in the bacteria or viruses. So here we try to find new targets which are not done before, which are not present before. So we try to find this kind of new targets, new enzymes in the bacteria and viruses. We work on that. And we also work on some undeveloped drugs, which left by companies because they don't have money or something like that. So we take that compounds from the companies and we try to develop them. So I don't want to go in details what different projects are, but just to give a brief overview. So there are currently 23 projects going on, which are spanning around these 5 research areas. Then what we want at the end from each of these research area, at least one compound can go into the preclinical trials. So that's what our goal is. Okay, enough about now what we do, what is drug discovery and what we do at GARDP. So dive deep into maybe science now. Let's talk about role of chirality in drug discovery. And I think this is maybe now chirality is taught at HSC level, right? Or in the first year of BSc. So all the students might know what chirality is, but just wanted to give you a brief overview. Chirality is a property of an object having both left and right-handed forms, right? They are called mirror images of each other. And if you try to superimpose them on each other, they are not superimposable, right? So this is called chirality. And we, in a chemical term, you know that, we call them as enantiomers, right? So just take an example. This is S and R form of this compound, which we call it enantiomers. And you all know these enantiomers could have different properties, right? So just take a day-to-day life example now. So you see this fan, which is running clockwise, which blades are clockwise direction, and this fan, which is an anti-clockwise direction, right? So if you start these fans and try to overlap on them, each other, they are not superimposable, right? So if you turn on them, so with this fan, the flow will go towards you. And in this fan, when you turn on, the flow will go, airflow will go away from you. So similarly, these compounds can also show different properties because they are arranged differently, right? So just take an example now, hypothetical interaction of these enantiomers in the active site of target. I told you about the target, which is enzyme. And this enzyme has this kind of cavities inside them, right? Where the catalytic reaction works. And so, for instance, the ATP generation, the energy generation in the bacteria and all. And this is catalyzed. So all the catalytic process are done in this kind of a void space. So just take an example of this S and R enantiomer. We try to fit it into this pocket, right? And you see that this enantiomer fit quite well. So there is no chance of the enzymatic process to go on in this space. So we can stop that enzymatic process. But if you look into this, it is not fitting well. So it's still there is still a chance that this enzymatic process can go. So here, using this compound might not be useful to kill the whole bacteria or virus, but using this compound, you can kill the bacteria or virus. So they are similar kind of compound, but just oriented differently. And you see that how different their interactions inside the target or enzyme would be, right? They also behave differently inside your body. So if you take one form of the enantiomer, it can absorb, distribute, or metabolite or excrete differently. And it can also shows a different effect on your body. So some effects, one of the enantiomer may be kill only bacteria, but the other enantiomer may kill even the human cells. So this kind of difference called pharmacokinetic pharmacodynamic effect, go through Google, search about it, you will know better about it. I don't want to give more details. So just give you an example, there is this called thalidomide tragedy, right? So this was the compound or the drug developed by a German pharmaceutical company called Grünenthal in West Germany. And it was marketed as a mixture of S and R enantiomer, right? So oops, sorry. Yeah. So it was introduced as a sedative drug, means to give you sedative effect in 1958. And it was proclaimed as a wonderful drug. It works on insomnia, like sleeplessness, cough, cold, headache. It was curing everything. And it was also useful for, as an anti-emetic drug. Anti-emetic means when you feel nauseous, when you feel like vomiting, right? So you take nowadays Ondem or something like that. So at that time, this drug was also used for this kind of purposes. And as you know, the pregnant ladies, they get this morning sickness. So at that time, the pregnant ladies took this drug to relieve this morning sickness symptoms. And what happened in 1950s and 60s, around 10,000 to 12,000 babies were born with the birth defect. They were born with the short limbs, short legs, and around 46 countries were affected with this drug. And the chances of survival of these babies was around like 40% of them were died during the first year of the birth. So really tragic situation. And so why it happens actually is because we found it out afterwards that this R enantiomer, which is called teratogen, was the compound which was giving this side effect, which was which was kind of interacting with a developing baby in your womb. And the S enantiomer was the one which was a real sedative, real drug. So nowadays, for any drug which want to come into the market, they have to test all these enantiomers, all the isomers of the drug, and check if they have some adverse effect. So you know what, now I think I convey you how different enantiomers can affect differently, right? But there might be a question how to get these different enantiomers selectively, right? And I think most of the master students, they know what is enantioselective synthesis or asymmetric synthesis, right? So how do we introduce this kind of chirality in the molecule? I don't want to go in details. If you want to know more about it, you can ask your teachers, your professors, and they will tell you more in details how this enantioselective process works, right? So this enantioselective synthesis, where you synthesize selectively one form of the drug, nowadays is applying in a late stage of drug discovery, in a lead compound generation, not really from the start. Why? Because it is complex reaction, and there are really scarcity of methodology, means there are really small amount of methodology which you can use to synthesize this kind of drugs. So that's why the companies don't want to spend more money to work on this complex reaction and develop thousands of compounds. If they can do it on few compounds, that's okay. That's economical. And what we wanted to do when I joined University of Groningen, to develop a reaction methodology which can synthesize this kind of chiral compounds in an easy way. So I was a part of this study where we develop a catalytic enantioselective synthesis of chiral heteroarenes through carbon-carbon bond formation reaction. So just give an example, the nitrogen-containing aromatic heterocycles, they have a significant biological importance. And out of 200 best-selling drugs in the world, 35% of them contains one or other heterocycle. And the majority of drug candidates means they are not drug yet, but they are in the preclinical trials. Around 90% of them contains one or more of nitrogen-containing aromatic heterocycle. And half of these drug candidates are chiral. So we thought, can we join heterocycle and attach a chiral center next to the heterocycle? So we know that chirality plays important role in a drug discovery, and also heterocycles are abundant, so just combine them. So we wanted to develop a general methodology to access chiral center next to the nitrogen-containing heteroarenes. So our aim was to do nucleophilic addition to this double bond, which is next to the heteroarene. So it looks like the substituted styrene, right? So instead of the heteroarene, if you have benzene here, it's a substituted styrene. So what we wanted to do is to do nucleophilic attack on the beta position of this double bond. But the main challenge here was it has really low reactivity of this double bond, right, towards the nucleophilic addition. If you compare it with alpha beta unsaturated ketone, where this double bond is in conjugation, so you can get 1,4 addition at this position, right? But in case of heterocycles, this localization of electron is not that good. So the reactivity is really low. So we search in the literature if there are some precedents before, and we found that there are 2 examples were reported. One is addition of a hydride, so just a hydrogen atom, hydride, right? So here there are limitations that you can only add a hydrogen atom. So you can't really grow a compound a little bit more complex. And the other one was a little bit better, which is carbon-carbon bond forming reaction, where you can add aryl boronic acids at the beta position, but it has also limitations that it can only add aromatic groups there. So there was no general methodology which can add aliphatic and aromatic groups at the same time. So what we wanted to do is to use a Grignard reagents, right? Everyone know now what is Grignard reagent. So we wanted to do copper catalyzed Grignard addition to the beta position of this double bond and having the heterocycle here. So there was only one example reported where this pyridine ring, that's parapyridine, was used, and you can see the enantiomeric excess was really low, 15%. We want around 90% of selectivity. So that was the only one example where they add phenyl magnesium bromide in presence of nickel and a chiral ligand. So that's a complex. So nothing really fascinating. So what we thought, let's try with benzoxazole and add ethyl magnesium bromide in presence of copper and a chiral ligand as a complex. And we run this reaction at -78 degrees Celsius and room temperature. So these were the ligands used and the different solvents we used. And we found out that at -78 degrees Celsius, no reaction. What that indicates that these compounds are really low reactive. And if we increase the temperature, there are a lot of side products, but no reaction, no addition of ethyl magnesium bromide on the beta position. So we know that we need to run this reaction at lower temperature, but we also want to increase the activity of this double bond. And the age-old technique, what Lewis acid is, right? Lewis acid tends to withdraw electrons, right? So what we thought, there is a lone pair of electron on the nitrogen. If we put the Lewis acid can pull the electrons, and with the resonance effect, the beta position can be more reactive for a nucleophilic addition. And it might help us, but there is also a problem. Lewis acid is acid, and ethyl magnesium bromide or Grignard reagents are base. So there is a compatibility issue. there might be a possibility that both of them will quench each other, will kill each other, right? And also, Lewis acid can also destroy the complex, the copper complex. So what we thought, let's try it. So we went in the lab, I put the reaction, I add ethyl magnesium bromide, and here I use Lewis acid, different Lewis acids. So TMS triplet, BF3 iterate, this titanium trichloride and TMS chloride, magnesium bromide, everything with this ligand, right? And to our surprise, we found that BF3 iterate was giving us really high yield, isolated yield after purification, and really high enantiomeric excess. So 96% of one form of compound was formed. So that was kind of a really good moment for us to find out that we tackled the reactivity of this compound even at minus 78 degrees Celsius. So what we thought at that time, that at minus 78 degrees Celsius, there was not enough time for ethyl magnesium bromide and Lewis acid to react together, but Lewis acid first reacted on the nitrogen lone pair, activate this double bond, and then ethyl magnesium bromide attacks on this beta position. So we do a lot of optimization, maybe in day-to-day practical labs, you do this kind of optimization. So we screen different ligands. You can see the ligand structures here, different solvents, different Lewis acid, and we found out that the BF3 iterate was the best Lewis acid. Ligand 1 is the best ligand, and diethyl ether was the best solvent. So this was the optimized condition for this reaction. Then we do some substrate scope. So what is substrate scope? We try to change this R group. So different phenyl groups here, different substituents on the phenyl group, and all works quite well. You can see that the yields were really good, and the enantiomeric excess, which was above 90% in most of the cases. In case of, instead of having this phenyl group, we also tried with just a methyl, and it works also well. Although the enantiomeric excess was a little bit low, but this is really good. Then we thought, why not to try other N nitrogen containing heterocycles? So oxazole works fine. Then pyrimidine works perfect. So it goes even 99% enantiomeric excess. Triazine works also fine, really good enantiomeric excess and isolated yield, and also quinoline. So that shows how general this methodology is. So you add a nitrogen containing heterocycle having a double bond next to it, and our methodology can give you one enantiomer selectively out of the other. So the only problem was with pyridine. Pyridine, we were not able to get the pyridine active. So I will come back to it after some times, how we'll tackle the low reactivity of pyridine. Then we also checked the Grignard scope. So we added, instead of ethyl magnesium bromide, different Grignard. So linear Grignard, branch Grignard reaction, Grignard reagents, and even the functionalized one, right? And all of them works quite good, really good enantiomeric excess and yield. We also tried the same Grignard scope on a different heterocycle, and it also works even better. So with the pyrimidine, we got enantiomeric excess of almost 99%. So 99% means we selectively produce only one enantiomer out of the other using this technique. So to summarize this work, we got Lewis acid and Grignard reagent combination to tackle the reactivity of the alkenyl heteroarenes, so which are low reactive compounds. Variety of chiral heteroarenes, as I showed you, with excellent yields and enantioselectivity can be accessed with this methodology. And there is a remarkable scope of Grignard reagents, including alkyl, aryl, linear, branched, as well as functionalized ones. And we were pleased to publish this innovation in journal Science, which is the reputed highest, like Science and Nature are the highest ranked journals in the chemistry. So we published that research into the Science. Let's going back to the pyridine story. As I told you, that we were not able to activate this double bond in the pyridine. So why it is so? They were intrinsically less active compounds other than the heterocycles. They have weak inductive effect in case of 3-alkenyl pyridine and 4-alkenyl pyridine, right? And in case of 3-alkenyl pyridine, there is not even a resonance activation, right? So in case of 2 and 4, there is a resonance activation, but with 3, it's impossible to activate this double bond having the Lewis acid attached to the lone pair of a nitrogen. Moreover, there is another possibility that this lone pair of the nitrogen or this pyridine, they are also good ligands for copper, right? So they might also decompose your complex. We are using copper ligand complex here. So that could be one of the possibility. So what we tried, let's change the Lewis acid from BF3, which is a little bit weaker, comparatively weaker, to TMS triplet. TMS triplet is really strong Lewis acid. So BF3 etherate was not working. So this way is not working. So we tried TMS triplet. And we found, this is NMR study, we found that this TMS triplet can form this adduct in between. So we usually get a really nice activation of this double bond and the beta position when we use a TMS triplet. And that is also at minus 78 degrees Celsius. So similar like last time, we use ethyl magnesium bromide, we use different solvents and different ligands with TMS triplet. So now we sought out which Lewis acid we wanted to use. So TMS triplet was the best Lewis acid for us. And here dichloromethane was the best solvent. And again, the ligand 1 was the best ligand. So this is how we tackle the reactivity issue of pyridine. So substrate scope, as I told you, the 4-alkenyl pyridine and 2-alkenyl pyridine works quite good. So we do, so having 4-alkenyl pyridine, 2-alkenyl pyridine, addition of ethyl magnesium bromide works really nice. It also works on a functionalized pyridines. So you have functional groups on that. And there's something really interesting here. Look at this ester group here. So Grignard reagent can also attack 1, 2 on this carbonyl position, right? But using our methodology, it is selectively attacking on the beta position. So that means that the Lewis acid is activating this position better as compared to the ester 1,2 addition. So we also have a selectivity when we use a combination of TMS triplet with the Grignard reagent. Similarly, we also do Grignard scope, so where we can add linear Grignard reagents. Even the methyl magnesium bromide, which is really problematic in some cases, works really well in our hands. We can also add branch Grignard reagents and also the functionalized ones. So after the functionalization, after the addition of Grignard, we can also functionalize these pyridines even further. So for instance, reduction of this pyridine, and then formation of this tetrazole group here, addition of this alpha beta unsaturated ester here, or all the functional groups on the pyridine that can also be accessed. And we also published this study in a really high-ranked journal called Nature Communication, and we were really pleased to put forward this kind of general methodology so that it can be used in general to develop a lot of compounds, and then maybe we hope that not only this methodology but also the academia, the basic research can find this kind of new reaction, new techniques where we can access this chiral compound really easily as compared to the known enantioselective reaction at the moment. And we hope that the use of these chiral compounds using these new methodologies can trigger the use of this chiral compound in early stages of drug discovery, for instance, in the hit compound generation and not in the late stage of drug discovery. So this is my outlook, and this brings me to the end of my talk. And before ending my talk, I just wanted to give you a brief overview of opportunities for the young and enthusiastic researcher. Shailesh already covered the different research grants you can apply. And I also put that here. Maybe you can ask me afterwards by email if you're really interested in. There is this website, you can also note it down. And there you can also find all the available research grants which you can use for doing your PhD or master's abroad. But before going that, what are the requirements? So I can say that I'm coming from Achalpur. I did my bachelor. I was really an average student, not really a bright student. So I did my bachelor from Achalpur. I did my MSc from Science, Shivaji Science College, Amravati. So if I can do it, I think anyone else can do it in the audience, all the students which are present there. So what is the requirement? So you need actually a high quality master's research project. At my time, we didn't have that. So we were not having this master research project. So what we did, because I got a chance to work in National Chemical Laboratory, and that was the gateway towards getting this kind of fellowship, right? So I asked if possible, the master students to go maybe in a bigger research institute, work there for a few months, and to check to do their master research projects. So not everyone can go to NCL or IICT, but maybe you can try to go to the university or Nagpur University, somewhere where you can do a better research. Not like whole research, but at least few months you can do that. Then you should know, you should have some experience in handling basic analytical instruments, GC, LCMS, NMR, and you can get that access to when you work in bigger institutes. So you should have basic knowledge of literature search, and I think nowadays you are doing that because you all have master's project to do in Amravati University. So publication in international journal is not required to get a PhD fellowship, but if you have it, it's better. So if you can get it one way or the other, better. So you should also kind of you should have a thinking that how my future research project would be. So you have to write a research project as well. Good academic record, it's not like it's mandatory. If you have a good project or good ideas, you can have like an average academic record as well. So having a good academic record is a plus point for sure. But that's if you have a average academic record like myself. I had an average academic record, but I had good projects in NCL and good exposure. So I was able to get my first PhD grant. So this is all about it. So today I would like to acknowledge Professor Harutunian and Professor Anna Hirsch. So because I just gave their name because I discussed about the research work which I did at University of Groningen and applied it into the Helmholtz Institute for Pharmaceutical Research. So that's why, but there were a lot of teachers. So all my teachers from my BSc in Jagdamba College and all my teachers from Sri Shivaji Science College, Amravati. So there I actually get involved in more organic chemistry and get more more knowledge for that for organic chemistry from all these teachers. So I also thank them, and of course the funding agency which generously fund my projects, University of Groningen, Helmholtz Institute, my Marie Curie grant from the European Research Council, which was really generous amount of money to fund my project. And I also would like to thank all the audience, all who listened my talk carefully, and organization committee once more to give me a chance to talk about my research work. And thank you all. So that's it from me.
I'm very much grateful.
I would like, hello, hello. Hello. I'd like to extend my warm welcome to our guest of honors, Honorable Professor N.S. Gadbye sir, ex-Doctor and ex-PC of TDU Gorakhpur University. I welcome Professor A.S. Aswar sir, ex-head of the Department of Chemistry in Santa Gadge Baba University, Amravati. I welcome Dr. Kodape ma'am, Associate Professor and Head of the Department of Chemistry in Santa Gadge Baba University, Amravati, and Senate Member and POS Member. I welcome Dr. Barabde ma'am, Associate Professor in Department of Chemistry, Santa Gadge Baba University, Amravati, and Senate Member. I would like to request the chairperson of today's program, Dr. Ramteki sir, to occupy their respective seats. Thank you, sir. I would like to request Professor N.S. Gadbye sir to occupy their respective seats. Thank you, sir. I would like to request Honorable Professor A.S. Aswar sir to occupy their respective seats. Thank you, sir. I request Dr. to occupy their respective seats. Thank you, ma'am. I request Barabda ma'am to occupy their respective seats. I would like to request Dr. Gavhadi sir, convener, HOD of Chemistry Department and IPSC coordinator of GS Tokyo Art, Commerce and Science College, and Dr. Bhrande sir, organizing secretary, to occupy their distinguished seats. Thank you, sir. I would request all the faculty members to occupy their respective seats. Thank you, everyone. We are privileged to have you among us today, and we thank you for taking time out of your busy schedule to extend your support, and your esteemed presence has indeed glorified this workshop and added vibrancy. Light made up in the darkness. Light symbolizes the knowledge and wisdom of free will. To solemnize this special invitation, I would like to invite all the dignitaries on dais. Okay. Flowers are our expression expressed to gratitude and respect. Hence, I would like to request Dr. Kawadi sir to welcome the chairperson of today's program, Dr. Ramteki sir, by offering a bouquet. Thank you, sir. I would like to request Dr. Nakari sir to welcome Professor N.S. Gadbye sir by offering a bouquet. Thank you, sir. I would like to request Dr. Udarande sir to welcome Professor A.S. Aswar sir by offering a bouquet. Thank you, sir. I would like to request Ingrid sir to welcome Dr. Kodape. Thank you, sir. I would like to request Chandan sir to welcome Dr. Barabde ma'am by offering the bouquet. Thank you, sir. Now I would like to request Dr. Gahadi sir to enlighten us with their words.
Good evening to all of you. Good afternoon, actually. Good afternoon to all of you. So I think this is the concluding session of two-day national workshop on modern approaches in material research. For this valedictory function, we have chairperson Dr. R.S. Rampati sir, chief guest Dr. Professor N.S. Gajviye sir, who is the president of NAFI, Dr. Professor Aswad sir, who is the advisor of NASI, Dr. Korape madam, Dr. Baramde madam, Dr. Ugarande sir, and other my collegiate colleagues, my students, and the students from other colleges. So actually, the idea behind this workshop was given by Dr. Aswad sir when he talked with me about organizing this workshop. Then instantly I realized that students from Chandur Bazar region or students from the rural region need an idea about the research culture, and to improve the research culture and to motivate and to accelerate their ideas about the research, we I personally uh, organized with the help of the NAPIC. We organized this workshop, and Dr. Aswan sir told me that we are organizing this workshop, and I didn't know the that whole nature of the workshop. That there are three plenary lectures, six invited talks. This is a huge program. This is a program like an international conference, but with the efforts of Dr. Aswar sir and Professor Gajviye sir, this program definitely becomes good for the students from the rural area. And I sincerely thank Professor Gajviye sir, who stay with us whole 2 days, and he enjoyed, I think, his part so much. And I think he was invited at an international conference at Nabira College, Patol, but he didn't went there. He stopped here, and his motive to stop was he want to interact with the students of the rural area. So I think uh, this program is hopefully successful. In these 2 days, we have different resource persons from Central University, State University, and CSI Laboratory. So I think this was a successful event due to your participation. And I also suggest you that you totally get involved in the teaching and learning all these processes, because this is the part from here you can uplift your life. As a last lecture I have listened, Dr. Ravi Dumre, he was also from a rural area, and he did his graduation from Achalpur and did PG from Sri Shivaji Science College, Amravati. And now he are associated with the Global Research Society. So this is one example. There are numbers of hundreds of examples from the rural area. So with this, I congratulate to all of you for uh, attending and participating in this workshop, and thank you very much.
मराठीमध्ये बोलतो. Good afternoon, sir. My name is Ganesh Sone from Government Reserve Institute of Science and Engineering, Ahmedabad. जेव्हा हे वर्कशॉप जेव्हा पावसाळ्यामध्ये आले तेव्हा आल्याच्या नंतर डिपार्टमेंटमध्ये एकच गोष्ट होती की आपल्याला कंपल्सरी या वर्कशॉपला जायचं, आता येतो मी क्लासमध्ये सांगितलं. परंतु मुलांचं कसं असतं, ऐकण्याची टेंडन्सी कमी आणि वर्कशॉप म्हणजे असतं काय, कारण की नवीन पिढीमध्ये एक आढळून येतं की मोबाईल जसा सुरुवात झाली मोबाईलला तेव्हापासून कुणाचं ऐकून घेतलं नाही, बरोबर आहे की? त्याच्यामुळं नवीन या ज्या इनोव्हेटिव्ह फंक्शन्स मध्ये सर्वात चांगला जो एक्सपिरियन्स असतो, जे एक्सपर्टीज असतात त्यांनी त्यांचा एक्सपिरियन्स आपल्यासोबत शेअर करतात. म्हणून मी सुद्धा या वर्कशॉप मध्ये रजिस्ट्रेशन करून पार्टिसिपेट होण्याचा प्रयत्न केला. तेवढं मधून सांगतो. थँक्यू.
Good afternoon to all the respected teachers, all the students. First of all, thank you for really talk because I'm doing modern transportation material research. This is our station, and sorry, it's a very fantastic what we did in the 2 days. We learn a lot of things we don't know about that. The new things we learn from this, and actually it was really nice for us. And thank you for arrange this session, and thank you for all of you.
Myself, Ajay Shada, student of PG Department of Chemistry. It was a great for to have the opportunity to attend the workshop. Thank you so much for the workshop. It was sorry, थोड़ा नर्वस तो मतलब फर्स्ट टाइम में बोलने का इसलिए थोड़ा सा नर्वस हूँ, बोल नहीं पा रही बराबर, बट मतलब यहाँ पर लोग यहाँ पर अपॉर्चुनिटी दी गई है, मतलब मैं उसके लिए बहुत खुश हूँ कि यहाँ पर मतलब अरेंज किया गया है, बहुत अच्छा एक्सपीरियंस रहा है और अच्छी इन्फॉर्मेशन मिली है. थैंक यू सो मच. Thank you very much. Now I would like to request Professor Abbas to share the
नमस्कार. गुड इव्हनिंग तुम्हाला नाव. कार्यक्रमाचे अध्यक्ष सन्माननीय रामटेके सर, समारोपीय कार्यक्रमाचे प्रमुख पाहुणे आदरणीय गजबीये सर, कार्यशाळेचे संयोजक आणि आमचे मित्र नंदकिशोर गवारे, सहसंयोजक विद्यार्थी श्रीकृष्ण कोडापे मॅडम, बराबदे मॅडम आणि आपल्या सगळ्यांमध्ये प्रसिद्ध माझे विद्यार्थी धर्मेंद्र दुपारे, इंगळे सर आणि महाविद्यालयाचे सर्व प्राचार्य आणि माझे प्रिय विद्यार्थी मित्रांनो, आपण सगळे विद्यार्थी वेगवेगळ्या कॉलेजमधून आले आहे. कोणी देवीचा आहे, कोणी विद्यार्थी आहे. तर सर्वप्रथम नाशिक चॅप्टरच्या वतीने आय वेलकम ऑल फॉर दिस कॉन्फरन्स फंक्शन ऑफ द वर्कशॉप. And I once again congratulate to convener
of this workshop, and my esteemed friend Gaware sir, Ramdev sir, and his entire management, as well as all the colleagues for their dedicated efforts, hard work, study, coordination, and each and everything. 2 दिवसापासून तुम्ही सतत ऐकत आहात. टोटल आपले 12 लेक्चर प्लॅन होते. 12 पैकी 11 सक्सेसफुल सगळ्यांनी ऐकलेले आहेत. काल टोटल 6 लेक्चर आपण ऐकले. बिगिन विथ द मॅडम टीका आचार्य, and she rightly pointed out how one has to develop an innovative idea with a simple and physiological effect, and how domestic lighter is one of our science discovery also. Then secondly, Dave sir also gave us the brief importance about nano fertilizer and how the old procedure are even today survive in the year of 2020. त्यांनी महत्व सांगितलं, परंतु देर इज ए वाईड स्कोप, तुम्हाला नोबेल प्राइज मिळवण्याचा वाईड स्कोप आहे. कारण अजूनही आपण अमोनियाची जी प्रोसेस आहे सिंथेसिस करण्याची, वी हॅव नॉट चेंज सल्फ्युरिक ऍसिड सुद्धा, बिकॉज स्टील टुडे वी हॅव नॉट गॉट हाय प्रेशर आणि हाय टेम्परेचर कन्झर्वेशन ऑफ एनर्जी असतो. परंतु त्यांनी सांगितलं की अराउंड 80% प्रोडक्शन ऑफ दिस थिंग डिपेंड ऑन. म्हणून तुम्हाला स्कोप आहे तुमच्या इनोव्हेटिव्ह आयडिया किंवा तुमच्या क्रिएटिव्ह थिंकिंगला वाव देण्याचा. How one can introduce new catalyst and new modified procedure आपण ऐकला शेवटच्या लेक्चर मध्ये. नंतर दवे सरांनंतर हितेश कुलरी यांनी गाईड किया की give the importance of nano drugs and drug discovery also, therapeutic drugs and particularly cancer drugs. Then in the post live session we have the esteemed speaker and dedicated teacher, outstanding researcher Professor Dhawale sir. And Dhawale sir has given the right message for all of us, not only we people also. And he has given the 4D formula: devotion, dedication, determination, as well as discipline. And I am sure all of you possess all these 4D with you. And I must congratulate you. Since last 2 days, I am observing you how you are disciplined way, attending continuously you there the bombardment of lectures. Sometimes the topic is so high, sometimes it was difficult. We people also, because not all topics are your concerns. सगळे टॉपिक आपल्या आवडीचे नंतर सगळे आपण शिकवत नसतो आणि सगळ्यांबद्दल माहिती पण नसतो. तरी तुम्ही खूप आवडीने ऐकत होते. दॅट रिफ्लेक्ट तुम्हाला आवड आहे. दॅट्स व्हाय यू कीप हिअर सच लॉंग डिस्टन्स. नंतर दिनेश कुमार यांनी सुद्धा ऍब्सॉर्बन्स सांगितलं आणि मेटल नॅनो पार्टिकल कसे इम्पॉर्टंट असतात मेथोडॉलॉजी त्यांनी सांगितले. राईट, हिअर युअर व्हेरी गुड आयडिया जातो. नंतर त्याच्यानंतर कमलेश श्रीवास त्यांनी सुद्धा सेन्सरचं महत्व सांगितलं आणि किती सिंपल रिसर्च आउटपुट तुम्हाला देऊ शकतो, किती सिंपल लुसिड वे सुद्धा. आणि हाऊ वन मेसेज दाखवला माहिती होते. हेल्दी माइंड हेल्दी बॉडी, आउटकम काय, हॅपी लाईफ आहे ना. म्हणून सगळे आपण 2 दिवसांपासून हॅपी कशामुळे आहोत, सरांनी आपली खूप चांगली व्यवस्था केली आहे ना. भोजन व्यवस्था सगळ्यात महत्वाची असते कुठल्याही कार्यक्रमाची. आणि कार्यक्रमाचं महत्व आणि इव्हॅल्युएशन, लग्न कसं झालं, छान झालं, जेवण सगळ्यात छान होतं. म्हणून 2 दिवस झाले सरांनी भोजनाची व्यवस्था छान केलं, पण कार्यक्रम सक्सेस झाला. परंतु जे श्रोते सुद्धा होते त्यांनी आपल्याला काय केलं, त्यांनी आपल्याला थॉट्स प्रोव्हाइड केले, फॉरवर्ड थिंकिंग. दे हॅव ऑल्सो प्रोव्हाइड फूड फॉर अवर थॉट्स. त्यांनी आपल्याला थॉट्स दिले विचार करण्यासाठी, आपल्या माइंडला थोडेसे कल्पकता वाढवण्यासाठी थॉट्स पण दिले. नंतर आज सकाळपासून आपण 3 छान लेक्चर ऐकले आहेत. इनोव्हेशन, क्रिएटिव्हिटी आहे ना. दोन्ही लेक्चर खूप सुंदर झाले अशा. आणि तुम्हाला मार्केटिंग कसं करता येते. आणि आपण फक्त बरेचदा काय होतं, सर प्रश्न विचारत होते पण तुम्ही पूर्ण बोलत होते. मी कसं म्हणतो की अजून आजपर्यंत तरी सगळे लोक युट्यूब बघतात, सगळे लोक युट्यूबवर जॉईन आहेत, सगळ्यांचे व्हिडिओ सुद्धा आहेत. पण तुम्ही आणि मी एखाद्या युट्यूबचा शोध लावला का, नाही लावला, कारण सगळे बाहेरचे आहेत. आपण विचारच करत नाही. काल सरांनी सांगितलंय 2+2 आपण कॅल्क्युलेटर काढतो. आज सकाळी सुद्धा सरांनी सांगितलंय, दीक्षित सरांनी, हम हमारे दिमाग को पानी देते थे, तुरंत क्लास में गुरुजी पढ़ाते हैं, हम तुरंत मोबाइल निकालते चेक करते रहते. वी नेव्हर थिंक डिफरंटली, आउट ऑफ बॉक्स आम्ही विचारच करत नाही. त्यांनी सकाळी आज छान आयडिया दिली आहे की इनोव्हेशन इन्फॉर्मेशन मध्ये काय फरक असतो आणि विच इज द नीड ऑफ अवर. या काळाची गरज आहे आणि रिसर्च हेच चांगला असतो विच हॅज रिलेव्हन्स टू द सोसायटी. समाजाशी त्याचा काही रिलेव्हन्स असलं पाहिजे, त्यालाच महत्व आहे. योगायोगानं आम्ही चांदूर बाजार कॉलेजची निवड यासाठी केली आहे. ग्रामीण भागातलं कॉलेज आहे, सिटी निगडीत कॉलेज आहे, सगळ्यांची असोसिएशन आहे आणि त्याचे ग्रामीण भागात आहे. विद्यार्थ्यांना एक चांगलं मार्गदर्शन मिळावं आणि तुम्हाला विद्यार्थ्यांसाठी एक थोडंसं फाट व्हावं ही याच्या मागची भावना होती. त्याच्यानंतर उमा शर्मा मॅडम सुद्धा छान लेक्चर दिला. मॅक्रोसायक्लिक मॉलिक्यूल राऊंड इथं त्यांनी एक्झाम्पल दिला. पोस्ट गेस फेनॉमिना संबंध, ठीक आहे. आणि त्याच्यानंतर पुन्हा कविता दीक्षित मॅडम इम्पॉर्टन्स ऑफ ऑल दिस थिंग एव्हरीडे लाईफ त्याचं महत्व काय असतं हे सगळ्यांनी दिलं आहे सर. तर म्हणून एकत्रित त्याच्यानंतर आपण 2 लेक्चर अगदी उत्कृष्ट लेक्चर पहिले लेक्चर थोडस किंवा ते एक्सलंट लेक्चर सिंपल कॅटलिस्ट त्यांनी दाखवलं. स्पिनिल कंपाऊंड तुमच्यापैकी बरेच मुलं करतात आणि ते सिंपल कॅटलिस्ट कंपाऊंड हाऊ ते यूज फॉर वॉटर प्युरिफिकेशन, टॉक्झिसिटी वॉटरमध्ये जे असतात, ऑर्गेनिक कंपाऊंड, ऑर्गेनिक कार्बन डिटर्मिनेशन करायला किती चांगले यूज करतात. म्हणजे कॅल्सिनेशन म्हणजे फर्नेसमध्ये हीट करणे फक्त कॉन्स्टंट टेम्परेचरला आणि रिएक्शन ऑप्टिमाइज करणे योग्य आहे. सो सिंपल रिसर्च वर्क पण त्यांनी कसे छान पब्लिकेशन दाखवले. समाजाची डिग्री करते रिसर्च वर्क आहे. त्याच्या शेवटचं लेक्चर अगदी उत्कृष्ट होतं आणि त्याने महत्वपूर्ण केलं की अचलपूरचा विद्यार्थी आज टॉप पोझिशनला आहे. म्हणजे तुम्ही सुद्धा तेवढेच टॅलेंटेड आहे. आज सकाळी सांगितलं कविता मॅडमनी सांगितलं आहे आणि दीक्षित सरांनी पण सांगितलं आहे की ऑल इक्वल टॅलेंट, फक्त फरक हाच आहे की कोणी त्याचा कसा उपयोग करतो. म्हणून त्या वर्कशॉपच्या माध्यमातून आम्ही तुम्हाला एक संधी उपलब्ध करून दिलेली आहे नाशिकच्या माध्यमातून आणि ह्याच्याकरता मी खरोखर वेदवीय सरांचे आभार मानतो, धन्यवाद मानतो. त्याला सपोर्ट केलं आणि आपल्याला कार्यशाळा करण्यासाठी त्यांनी मान्यता दिली आणि सरांनी आम्हाला ओपन ऑफर दिली की भविष्यात कुठलेही कार्यक्रम विद्यार्थ्यांसाठी करायचे असेल तर आय एम ऑलवेज यू डोंट बॉदर अबाउट जस्ट यू प्लॅन सिस्टमॅटिकली बट आय नीड ए सायंटिफिक प्रोग्राम. आय डोंट नीड होता है चलता है कर लेंगे. त्याच्यामुळे तुम्ही ऑब्झर्व केलं असेल सर त्याचे सिन्सिअरली होते. यू आर ऑल द वाईज प्रेझेंट इन द हॉल एक्सेप्ट फॉर 2 मोमेंट्स पण हिच रिफ्लेक्ट आणि ह्या गोष्टी सुद्धा शिकायच्या असतात. लोकांनी त्यांचं प्रेझेंटेशन कसं दाखवलं, पीपीटी कसं तयार केलं होतं, ते बोलताना कसे बोलत होते, ह्या गोष्टी शिकायच्या असतात, अनुकरण करायचे असतात, नुसतं ऐकायचे नसतात. आपण काय करतो वापर करतो, इथून ऐकतो इथून सोडून घेतो. विद्यार्थ्यांनी तसं करायला नाही. यू हॅव व्हेरी ब्राईट फ्युचर, यू हॅव व्हेरी गुड अपॉर्च्युनिटी इन युअर करिअर प्रोव्हाइडेड यू शुड बी डिटर्मिन्ड, यू शुड हॅव ए फिक्स गोल आणि गोल अचीव करण्यासाठी काही सिस्टमॅटिक प्लॅन लागतो आणि तुमच्यासाठी रामटेकेसर सारखे प्राचार्य उत्कृष्ट प्राचार्य तुम्हाला लागले आहेत. त्यांना बरोबर विजन आहे. अध्यक्ष विजयभाऊ टोपे आहेत, दादासाहेब आहेत, गवाडेसर सारखे उत्कृष्ट शिक्षक तुम्हाला लाभले आहेत. तुम्ही या लोकांचा सगळ्यात फायदा करून घ्यायचा. लोकांचे सुद्धा खूप चांगले संबंध आहेत. म्हणजे कोडापे मॅडम, गुरे मॅडम, बोथ ऑफ देम आर व्हेरी गुड टीचर अँड ऍक्सेसिबल पर्सन. त्यांचं मार्गदर्शन घ्यायचं. जिथे जिथे संधी आहे तिथे आपण जायचं, पण जावं लागेल नाही तर मी कशाला होत नसतो. तुमच्या नेहमी तुमच्या घरी कोणी येणार नाही, तुम्हाला जावं लागेल. म्हणून त्या वर्कशॉपच्या माध्यमातून, त्या कार्यशाळेच्या माध्यमातून मला खात्री आहे की तुम्हाला काही ना काही नक्कीच फरक पडला असेल आणि तुमच्या उद्यापासूनच्या रुटीन मध्ये जे असेल तुम्ही त्याला नक्की रिओरिएंट करणार. आणि जसा शेवटच्या डेंगू सरांनी सांगितलं की हाऊ यू मस्ट फ्रॉम सिम्पल विथ ऑर्डिनरी बॅकग्राऊंड ते एक्स्ट्रा ऑर्डिनरी, फक्त आपल्यापेक्षा थोडंसं वेगळं केलं तुम्ही सगळेच करू शकता सगळ्यांनी. म्हणून त्याच्यापासून घ्यायचं. आमच्याकडे डिपार्टमेंट होते हुसेन तो सुद्धा वर्किंग they have position also. Then we positive are. These are the very good job. Always ready. मला फोन करतात म्हणून येऊन दे ICT त्यांना फोन करतात. सर चांगले पोर असेल आम्हाला 31,000 पर मंथ फोन करतात. चांगले पोर असेल रिकमेंड करा. आम्ही विचार बोलला ना तुला पहिले डोक्याला हात लावणार त्याला सांगतो रिसर्च सांगितलं पण काळाची गरज आहे. सो यू शुड परफॉर्म विथ युअर नॉलेज, विथ युअर नॉलेज द सोसायटी असतो. आणि अल्टिमेटली थ्रूआउट सगळ्यांचे लेक्चर सोसायटी ओरिएंटेडच होते. सगळ्यांनी जे समाजाची निगडीत होईल आणि त्याच संशोधनाची आज आपल्याला गरज आहे. आणि हे सगळं करत असताना काल सुद्धा मी सांगितलं की 3 प्रकारचे लोक असतात. आज सुद्धा 3 प्रकारचं मॅनेजमेंट असतो. मी तेवढं तुम्हाला सांगतो. मॅनेजमेंट 3 प्रकारचं सिंपल असतो. सगळ्यात सोपं म्हणजे मटेरियल मॅनेजमेंट. सिम्पली सरांनी ऑफिसमध्ये बसून सांगितलं खुर्च्या उचला, टेबल उचला, इथं लावा तिथे लावा. दॅट इज इनफ. दॅट इज कॉल्ड एज मटेरियल मॅनेजमेंट. त्याच्यापेक्षा थोडंसं अबव मॅनेजमेंट असतो मनी मॅनेजमेंट. एकदम जरा 10 लाख दिले तर सर त्याचं मॅनेजमेंट करू शकणार नाही. 10 लाख एकदम आपल्याला मिळाले कसं करायचं. बट इफ यू प्लॅन in a differently. You will manage money management also. आणि कुठली मॅनेजमेंट असते ते तुमच्या आमच्याचं पीपल मॅनेजमेंट आणि हे पीपल मॅनेजमेंट गव्हर्नन्स आपण सगळं केलेलं आहे. सगळ्यांना 2 दिवसापासून त्याने लिहून ठेवलं आहे. म्हणून पुनश्च मी एक नाशिकच्या अभिनंदन करतो. आपल्या ग्रामीण भागातील विद्यार्थ्यांसाठी ही वर्कशॉप न करता इतर दुसऱ्या प्रकारच्या ऍक्टिव्हिटी आम्ही सदैव तुम्हाला मदत करू. कुठल्याही प्रकारच्या लागू अकॅडमिक असो, फायनान्शियल असो, रिसोर्स पर्सेंटेज असो, तुम्ही वर्कशॉप जरी गेले तरी मदत तुम्हाला करू. असं काही नाही फॅमिली मेंबर असो. म्हणून कुणाचे रामटेके सरांचे संपूर्ण अध्यक्ष सचिव सर्वांचे मनापासून आभार मानतो की त्यांना नांदीला संधी उपलब्ध करून दिली कार्यशाळा आयोजनासाठी. संधीच उपलब्ध करून दिली नाही, स्वतः विद्यार्थ्यांना जेवण सगळं दिलं, चांगली उपस्थिती दिली आहे आणि तुम्हा विद्यार्थ्यांना चांगलं फूड मिळालं आहे. तो अधिक वेळ न घेता, वेळ संपला, बरेच लेक्चर आहेत, हार्दिक पूर्ण झाले. अधिक वेळ न घेता सर्वांचे आभार मानून आणि गव्हर्नन्सचे मनापूर्वक आभार मानतो. सरांना मला एवढा सन्मान दिला, आदर दिला आणि आपल्या होण्याचा एक मान दिला, होण्याचा एक मान दिला, त्यासाठी सरांचे गव्हर्नन्सचे संपूर्ण आभार मानतो. धन्यवाद आणि शुभेच्छा देतो.
आता आपण आपल्या प्रोग्रामसाठी शेवटचा प्रोग्राम ठरलेला ह्या प्रोग्राममध्ये त्यातून काय शिकला, त्यातून काय योजना तुमच्याबरोबर आणि नंतर काय करणार हे याची तुमच्याबरोबर. आम्ही आमचं काम केलं, आम्हाला जे करायचे असेल जेव्हा मी त्यांना विचारलं, म्हटलं मला एक रुरल भागात प्रोग्राम घ्यायचा आहे आमच्या नाशिक नॅशनल अकॅडमी ऑफ सायन्स इंडिया. तर म्हटलं खेड्यातील किंवा सेमी रुरल अर्बन सेमी अर्बन एरियातील काही कॉलेज म्हटलं तिथे भोजपर मुलं असतील, केमिस्ट्री, फिजिक्स, बायोलॉजी आणि फिजिकल एनी अदर सायन्सेस, तो त्या मुलांमध्ये मला आपल्याला लेक्चर्स द्यायचे. तर सांगितलं मला पाहिजे BSc किंवा MSc ची किंवा पोस्ट ग्रॅज्युएट करणारे, पर्टिक्युलरली रिसर्च असलं तर नसलं तरी काय, त्याने मला भरपूर हे कॉलेज त्यांनी विकून दिलं. आणि ते टोमटे यांच्या छत्रछायाखाली आम्ही हा प्रोग्राम घेतला. तसेच आजच्या कॉलेजची आमची प्रिन्सिपल रामटेके साहेब प्रिन्सिपल परमिशन दिली. तसेच ह्या कॉलेजची काही टीचर्स फार हुशार टीचर्स मी म्हणतो त्यांना, आणि व्हेरी ऍडमिनिस्ट्रेटिव्ह आम्हाला सपोर्ट झाला. आपले गवाळे सर आणि उबर्डे येस ओके उबर्डे ह्या लोकांनी पासून बेसिकली मॅडम मॅडम आहे ओके. आणि आमच्या प्रक्रियाच्या शेवटच्या सेशनला आमच्या मॅडम बुढा अमरावती I think I'm satisfied as far as this side is concerned. Okay, I'm very happy to have this program here, and maybe in future, what these students want, these guys they want, tell me. I there is a program here. What they want, what your customers want. Okay, I don't want to do your choice, but their choice. Okay, we will do. So next time. तुम्हाला लोकांचे काय चॉईस असणार मला सांगा तुम्ही.
मला तेच पाहिजे होतं तुमच्याकडे फीडबॅक. पुढच्या वेळेस तुम्हाला काय शिकवायला लागेल ते सांगा, तुम्हाला काय आवडते. तसं आम्ही शिकवतो. आमच्याकडे खूप लोक आहेत, चांगले मोठे मोठे टीचर्स आहेत भारतातले, तुम्हाला शिकवायसाठी तयार. फक्त त्यांना आम्हाला त्यातलं तेव्हा दिसायला पाहिजे. आता त्याची व्यवस्था माझ्याकडे सोडली. त्या लोकांना सगळ्यांनी येऊन द्यायचं तुमच्यासाठी करायचं. आणि तुम्ही इंटरेस्ट घेऊन पण ऐकायचं, मग क्वेश्चन आन्सर विचारायचं. तो भाग कमी पडला तुमचा, क्वेश्चन आन्सर. पुढच्या वेळेस क्वेश्चन आन्सर इतके तयार करून ठेवा, कुठल्याही प्रोग्रामला क्वेश्चन आन्सर झाले पाहिजे. मला इंटरॅक्टिव्ह प्रोग्राम पाहिजे तुमच्याबरोबर. कुठल्याही टीचरने शिकवलं या इथे आपल्या प्रोजेक्ट असतील तर त्यांना तुम्ही क्वेश्चन आन्सर विचारले पाहिजे. तेव्हा मला खरंच सॅटिस्फॅक्शन होतं. कारण मी टीचर आहे ते काम करून आमचे स्टुडंट क्वेश्चन नाही विचारत, आम्हाला चॅलेंज नाही, तो नाही आम्हाला काय झालं तुमच्या गोष्टी काही गडबड झाली आपल्या लेक्चर असं वाटायचं. मग गुरु राहिली पाहिजे की तुम्ही क्वेश्चन विचारता, आज क्वेश्चन त्याप्रमाणे विचारला पुढच्या वेळेस काय विचार काय असं झालं पाहिजे. तुमची क्वेश्चन आन्सर फार इम्पॉर्टंट. कारण कुठल्या प्रोजेक्ट यंग जनरेशनवरती अवलंबून आहे. कारण तुम्ही आमची घरोवर चालवणारे लोक आहात. त्यामुळं माझी इच्छा आहे की प्रोग्राम आपण आता मी ठरवून टाकलं कुठे मी जर भागातून घेणार. ओके, जवळपासच्या कॉलेजमध्ये कुठेही घेईलच मी. तसे जवळपासचे कुठलेही कॉलेज नाही बघा तिकडे घेऊ. आणि तू डॉक्टर तुमचा सगळ्या पिढीला आपल्या त्याच्याकडनं फीडबॅक घेऊन त्याप्रमाणे प्रोग्राम अरेंज करू. ठीक आहे. आणि दुसरं म्हणजे मला आजच्या कार्यक्रमात जसे की तुम्ही पाहला कार्यक्रम वाढत कालपासून सुरू झाला, आता म्हणजे वाढत गेला. हे जे लेक्चर्स आलेले आहेत ते लेक्चर्स 10-15 दिवसांनी ठरवले. त्याला मला याला दिला आस्वादला हे 10-15 दिवसात त्यांनी एवढे चांगले लेक्चर त्या लोकांना बोलावून घेतले त्यांच्याकडनं. आय थिंक दॅट इज माय हॅट्स ऑफ दिस मॅन. So thank you. Naturally, attention supported in doing this program. Okay. तुमच्या प्रोग्राम मी पाहत होतो सगळं मी प्रत्येक लेक्चर हा बसलो आहे. प्रत्येक आमचे जे अकॅडमिक लोक असतात सगळ्यांना कुठल्याही म्हणजे बरेच माझ्या जीवनात बरेच नोबल अवॉर्ड पाहिजे त्याच्यावर बोललाय बसलो आहे. आमच्या प्रोग्राममध्ये कुठलेही येतात कधीही जागा सोडत नाही. पहिल्या बसले खुर्ची ते शेवटच्या क्षणापर्यंत असतात. आम्हाला बोर होत नसेल बसतो काय काय. आणि ह्यातले सगळ्यात महत्वाचं म्हणजे शेवट भाषण ऑनलाईन फार चांगलं छान बोलायचं त्या भागातलीच बोल आहे ती. आणि पर्टिक्युलर शेवट नाही सांगतो स्टोरी आहे जुमडे त्याप्रमाणे कमाल केली. त्याप्रमाणे असे जेव्हा 10 पेपर जरी काढले, सायन्स हे एक जर्नल आहे, जगातलं सगळ्यात मोठं जर्नल आहे. पण 10 जरी पेपर काढले तरी त्याला लगेच नोकरी IIT कानपूरला मिळेल असे 10 काही पेपर काढा सायन्स मध्ये किंवा नेचर. ह्या 2 जर्नल हे 2 नेचर लक्षात ठेवा. तेव्हा बरेच फोकस जर्नल बघता जरूर तुम्ही नेचर जर्नल वाचा आणि दुसरं काय ते फोर फ्रंटच्या न्यू सायन्स मध्ये आणि त्यातूनच बरेच मोठे नोबल निघतात. ओके, रिव्ह्यू दिस माय सेंटिमेंट. आणि त्यामुळे दोन्ही जर्नल तुमच्या लायब्ररीत आणा किंवा नसेल तरी वाचा. ऑनलाईन अवेलेबल आहे जर्नल. ओके, फार चांगले जर्नल्स आहेत. आणि आपल्या भारतामध्ये कुठलीच स्कीम नाही आहे, काहीच नाही तुम्हाला अवेलेबल आहे की. पण असे जर्नल तुमच्या तर तुम्ही मागवून घ्या असेल सोय. ओके,
हे दोन्ही जनरल फार महत्वाचे. ठीक? तुम्हाला फोर फ्रंट सायन्स ज्याला म्हणतो, आज काय चाललं जगात असं सायन्स तुम्हाला शिकायचं असेल, हे दोन जर्नल्स असतात. तर बाकी बाकी पण जर्नल्स खूप छान आहेत. पर्टिक्युलर फील्ड मध्ये बायो मध्ये पण सेल नावाचं जर्नल येतं. सेल बायो सायन्स मध्ये किंवा इंजिनिअरिंग सायन्स मध्ये IEEE जर्नल्स आहेत. छान स्टँडर्डचे आहे. अशा जर्नल्स मध्ये तुमच्या बाबतीत जगाला कळतं ह्या जगात कुठेतरी एक पोरगा आहे ते पब्लिश करणार, त्याला ते पकड सांगावं लागत नाही तुम्हाला. पब्लिश होते तुम्हाला आमच्याकडे या. त्या मोठे मोठे लोक त्यांना माहीत झालं की ते तुम्हाला कॉन्टॅक्ट करतात. म्हणून अशा ठिकाणी जर्नल वाचत तुला थोडं पाहिजे काय करायचं स्टाफ सर्कल ऑलरेडी पण ट्रक हे जे आहे सुरुवातीचे काही काम दिलेले यंग लोकांनी. ओके, राम डॉक्टर दवे ते आपले श्री and then Dawdi Saab. Dawdi Saab had starting the subject, what is called is the carbohydrate chemistry. Okay, in chemistry journal. Materials मध्ये there are journals like advanced materials. This is something very big journal. Physics मध्ये physics review letters. हे नाव सांगण्यासाठी की हे जर्नल्स मध्ये ज्यांनी त्यांनी पेपर पब्लिश केलेले आहेत ते लोक जगाचे फार मोठे मोठे लोक होऊन गेले. ओके, त्यामुळे अशी जर्नल वाचा आणि त्यापासून ते काय बोलतात काय तिथे लिहितात ते वाचलं तुम्ही. आमची टीचर आहे, आम्ही जोरानं बोलतो पाठतो तर ते त्या जर्नल्स वाचा तुम्ही, ते शिका, त्यात परत शिकायला मिळतं. फोर पण आयडिया येतात. तुम्हाला सांगतो पर्टिक्युलरली ज्यांना फिजिक्स करायचं असेल त्यांनी फिजिक्स रिव्ह्यू लेटर्स नावाचं एक छोटंसं जर्नल येतो, ते हमेशा वाचा. ओके, आणि त्या जर्नलचा तुम्ही वाचाल तर तुम्हाला फिजिक्स मध्ये काय काय नवीन नवीन सब्जेक्ट नवीन नवीन आयडिया इंट्रोड्यूस झालेले असतात त्या सगळ्या येतात. ओके, आणि बाकी जर्नलचे जर्नल्स वगैरे असतात. पर्टिक्युलरली मला लेक्चर्स आवडलेले शेवटचे लेक्चर आवडतात आणि बाकीचे पण चांगले आहेत, पण ते थोडे काही ऍडव्हान्स लेवलचे होते, तुमच्या बियॉन्ड होते काही. आमचा प्रयत्नाचा पुढचा राहील की तुम्हाला तुमच्या लेवलवरती लेक्चर पाहिजे, मुळातून बेसिक झालेली पाहिजे. त्या बेसिक मधनं कसं कसं आपण समोर जातो पाहिजे. कारण तुमची तुमच्या लेवल तुम्ही BSc ची मुलं आहेत मला वाटते आणि डिफरंट सेंटरचे मुलं आहेत. हा डिफरंट. हा अच्छा तर त्यामुळे असं वाईड स्पेक्ट्रम बरोबर ते बॅकग्राऊंड केलेलं वाईड स्पेक्ट्रमला सगळ्या लेवलची मुलं आहेत. पण क्वेश्चन आहे कोणी दिले नाही त्यामुळे काही कळायला लागलं नाही ना. झालं पाहिजे तुमच्यामध्ये, तुमच्याकडनं रिफ्लेक्ट झाला तर मला कळते की बरोबर ह्या लेवलचे प्रश्न विचारले आपण आणि काहीतरी काय त्या मुलाला समजतं म्हणून आहे ना. त्याप्रमाणे मग आम्हाला लेक्चर प्लॅन करायला ऑटोमॅटिक मिळतं, चालना मिळते. आम्हाला विचार लागत नाही तुम्हाला तुम्ही आता क्वेश्चन विचारायला शिका हा समस्या वेळेस. हा आता एवढंच आहे त्यामुळे मी सगळ्यांना I just thank you all for listening us, listening my kind of invited talk, and Dr. Rajwan and his Namte Sahib and his team. Okay, on this kind of occasion. Thank you very much. Thank you very much. Okay, thank you.
Now,
विद्या वैभव आणि घराण्याची ही पूर्वपरंपरा नसताना समाजासाठी शिक्षणाचं समुदाय विधायक कार्य करणारे वैराग्य गाडगे बाबा या चांदूरबाजारच्या नगरीमध्ये उच्च शिक्षणाची रंग आणणारे माझे आजोबा सगळी भूमिका विद्या संकल्पना प्रदान करणारी आई सरस्वती या व्यासपीठावर या 2 दिवसीय तुमच्या चर्चासत्राच्या निमित्ताने उपस्थित असलेले आदरणीय डॉक्टर गजबी सर, डॉक्टर कसवाल सर, आदरणीय डॉक्टर गुलाब मॅडम, डॉक्टर बारावली मॅडम, आमच्या या कार्यशाळेचे आणि आमच्या महाविद्यालयाचे IPSC कोऑर्डिनेटर आणि रसायनशास्त्र विभागाचे विभाग कवाडे सर, डॉक्टर रुग्णांचे सर आणि आमच्या महाविद्यालयाचे उपस्थित माझ्या महाविद्यालयाचे सर्व अध्यापक, माझे बंधू आणि माझे सगळे या ठिकाणी उपस्थित विद्यार्थी, मित्र, मैत्रिणी, बंधू. मला वाटतं की आमचं ग्रामीण महाविद्यालय आहे आणि आमच्या महाविद्यालयामध्ये वर्षभरात संत गाडगेबाबा अमरावती विद्यापीठात त्याच महाविद्यालयामध्ये जेवढे कार्यक्रम वर्कशॉप वगैरे नसतात तेवढे कार्यक्रम आमच्या महाविद्यालयात आणि मला वाटतं त्या निमित्ताने आमच्या विद्यार्थ्यांना यातनं वेगवेगळे ज्याला आपण रिसोर्स पर्सन म्हणतो की ज्यांच्या माध्यमातून तुमच्यापर्यंत वेगवेगळ्या पद्धतीचं ज्ञान पाठरायचं असतं आणि शेवटच्या घटकापर्यंत पोहोचवायचं असतं ही जबाबदारी आमची सगळे प्राध्यापक असतात आणि त्या निमित्ताने 2 वर्षीय चर्चासत्र ज्याला आपण म्हणतो सत्र आणि त्या चर्चासत्रामध्ये उपस्थित असलेले की ज्यांनी अतिशय सुंदर मला वाटतं की आता रसायनशास्त्रावर विषयी मी भूगोलाचा प्राध्यापक आणि मॅनेजमेंटचा म्हणून या 2 दिवसाच्या कार्यशाळेच्या निमित्ताने आपल्याशी काय संवाद साधायचा डॉक्टर कॅटेरिना पाहुणे इथून निघाले त्यांना करून आलो त्यांनी मला सांगितलं की या ठिकाणची जी परंपरा आहे आणि या ठिकाणी आम्हाला आनंद मिळाला तो आनंद की ज्या महाविद्यालय निर्माण झालेलं आहे की माझ्या परिसरातील गोरगरीब विद्यार्थ्यांना घरच्या या ठिकाणी त्यांना शिक्षण मिळावं त्यांच्या मनामध्ये आहे आणि त्यांनी या महाविद्यालयाची स्थापना केली की माझ्या त्याला विज्ञान त्याचं शिक्षण करतात या माध्यमातून स्कूल आणि एवढं शिक्षणाचं समाज ज्या ज्ञान आणि आज मी सकाळचं लेक्चर सरांचं ऑनलाईन देत असताना आणि त्यांनी मला इनोव्हेशन इन्व्हेन्शन त्यांची बुद्धी त्यांना पूर्ण ज्ञान दिलं त्यांना त्याला आपण निष्पत्ती म्हणजे ग्रामीण भागात तुमच्या मनात की स्वामी विवेकानंद म्हणजे विश्वास की बाहेरच्या जगामध्ये जे सुद्धा ज्ञान आहे ते ज्ञान काही त्याच्यामध्ये काही उपजत नव्हतं त्यांना जाणीव म्हणजे आपण जर उदाहरण घेतलं की सर आयझॅक न्यूटन यांनी शोध लावला
म्हणजे गुरुत्वाकर्षणाच्या नियमाचा शोध लावा म्हणजे गुरुत्वाकर्षणाचा नियम त्याच्या पूर्ण अस्तित्वात नव्हता किंवा आज अस्तित्वात नाही असं नाही आहे. तर फक्त एक झाडावरून पडणारं फळ त्यांनी बघितलं, ते झाडाखाली डुबके होते, त्यांच्या मनामध्ये त्या फळाविषयी एक चेतन बाधिता सुरू झाली आणि हे झाडावरनं पडलेलं पण खाली जमिनीवरच का आलं हे विचार चक्रांची प्रवाहित झालेली मानता आणि त्याच्यातून निर्माण झालेला तो एक सिद्धांत जो गुरुत्वाकर्षणाचा नियम म्हणून संपूर्ण जगामध्ये ओळखला जातो. तर विश्वाचा अनंत घटनामध्ये तुमच्या मनातील विद्यमान आहे आणि माझ्या जगात तुम्ही त्यांचे अध्ययन करण्याची संधी आहे ही भूमिका घेऊन प्रत्येक विद्यार्थ्यांनी काम करायचं. त्यांनी मला सांगितलं की इनोव्हेशन सेंटर निर्माण करा, म्हणजे तुमच्या मनामध्ये ज्या ज्या छोट्या छोट्या गोष्टी येतात त्या गोष्टी कुठेतरी कागदावर मांडा, त्याच्यातूनच इन्व्हेन्शन झालं, त्यामुळे इनोव्हेशन शोध लागलेला आहे. म्हणजे तुम्हाला म्हणताय की आता फक्त माझे लांब किती आहे की माझ्या मुलासोबत काल रात्री 12 वाजता धावलीला आहे तो खेळत होतो आतमध्ये त्याला सुट्टी होत्या बॅडमिंटन आणि वारंवार माझी केस माझ्या डोळ्यासमोर येते खेळताना बॅडमिंटन खेळत असताना. मुलींना नेहमी प्रॉब्लेम येतात प्रवासामध्ये जात असतात, तर त्यांच्यासाठी सेफ्टी पिन निघाली. आपण विचार करूया तर ही सेफ्टी पिन कशासाठी निघाली त्याला आपण म्हणायचं म्हणून डोक्याला आपण पिन असते कशाच्या निघाली. कुठल्या तरी त्यावेळेला अडचणी निर्माण झाल्या, माझे केस सतत बार होऊन जाईल आणि मला वाटते तिथल्या आयच्या करून दिल्या माझ्या केसांना आवडा आणि बॅडमिंटन त्याच्या टेबल खेळायला एक स्टेटमेंटचा पॉइंट कागदा भूमीला इनोव्हेशन काय आहे. कागदाचा घटना आहे, घटना हस्तबद्ध झालेला आहे आणि तुम्हाला एकत्रित करून त्या स्टेटमेंटचे निघितल्या. पण हा संपूर्ण जगामध्ये त्या स्टेटमेंटची किती महत्वाची विचार आपण कधी केला का. ते इनोव्हेशन आहे, ते इन्व्हेन्शन आहे, कोणाच्या तरी डोक्यामध्ये कल्पना आहे. सेफ्टी पिन लावायची आहे बा, ती सेफ्टी पिन सुद्धा तुम्ही विचार केला का ती इन्व्हेन्शन आहे, इनोव्हेशन आहे. म्हणजे तुमच्या मनामध्ये ज्या कल्पना येतात त्या कल्पना कुठेतरी कागदावर रेखाटल्या गेल्या पाहिजे, त्याच्यावर चिंतन मंथन झालं पाहिजे आणि त्या माध्यमातून आपल्याला सुचलेली कल्पना लोकांपर्यंत पोहोचून त्याच्यात काय अडचण निर्माण होतात आणि ती एखादी अडचण सोडवण्याचा प्रयत्न करणं इन्व्हेन्शन आहे मला असं वाटतं. आणि त्या निमित्ताने या ठिकाणी जेवढे रिसोर्स येतात त्यांच्या चेतनातून त्यांच्या बंधनातून तुमच्यापर्यंत एखादा विचार पोहोचणं, तुमच्या मनामध्ये एखाद्या विचारचित्रांची मालिका प्रवाहित होणं आणि त्या निमित्ताने एखादा विचार त्याच्यातून एखादा इनोव्हेशन किंवा इन्व्हेन्शन निर्माण होणं हेच खऱ्या अर्थाने विज्ञान. आणि या विज्ञानाच्या अनुषंगाने आज जगातील सगळे शास्त्रज्ञ जे काम करत आहेत म्हणजे तो एखादा मोठा स्फोट ज्याला आपण त्याची जगाची उत्पत्ती कशी झाली, अणु रेणूंच्या कुठल्या स्फोटातनं काय निर्माण झालं, हे पृथ्वीचा अंतरंग, हे पृथ्वीचा अवकाश, पृथ्वीचा रंग, आकाशाचा रंग निळा का पाणी निळं का, सीवी रंगने लावलेला शोध, या सगळ्या गोष्टी तारे का लुकलुकतात, सूर्य ग्रहण चंद्रग्रहण का होतं, या सगळ्या गोष्टींचा विचार तुमच्यासोबत होत आहे. तुमच्यासोबत तुम्ही पुस्तकामध्ये सगळं वाचता, पण तुम्ही त्या गोष्टींचा विचार करता का. प्रकाश तरंग विशिष्ट पातळीमधून गेल्यावर त्यांनी पाण्याचा रंग निळा दिसला, आकाशाचा रंग निळा दिसला. टीव्ही रमणच्या मनामध्ये त्यावेळेला आला आपण त्याचा विचार केला का आपण विचार होणार. ही भूमिका जेव्हा आपल्या मनामध्ये निर्माण होईल तेव्हा खऱ्या अर्थाने आपण विज्ञानाचे विद्यार्थी आहोत आणि आपण काहीतरी इन्व्हेन्शन करू शकतो. त्यांनी मगाशी पण मी त्यांचं लेक्चर सांगितलं की तुम्ही ग्रामीण म्हणले आहे तुमच्या मनामध्ये ते सतत सुरू राहिलं पाहिजे. तुम्ही प्रत्येक प्रॉब्लेमचं सोल्युशन करण्याचा विचार केला पाहिजे. प्रॉब्लेम धरून ठेवायचे नाही तर प्रॉब्लेम सॉल्व्ह कशी होईल याचा विचार तुम्ही करा. त्या दिवशी खऱ्या अर्थाने आपल्याला त्यांचं इन्व्हेन्शन इनोव्हेशन पण आणि तीच भूमिका घेऊन मला वाटतं विज्ञानाच्या विद्यार्थ्यांनी आपल्याला पुढे जायचं. मग ते आपण रघुनाथ भास्लेकर म्हणा की संगणकाचे विजयराव भटकर म्हणा, हे सगळे लोक की ज्यांनी जगाला संगणकाची एवढी मोठी सुपर कॉम्प्युटर दिली, पुरवण्यासारख्या एखाद्या छोट्याशा गावामध्ये जितकी लोकसंख्या दिल्लीच्या नव्हती नव्हती इथून ते निर्माण झाले मोठ्या जिल्ह्यात. तर आपल्याला हे सांगायचं आहे की आपल्या समोर जर तुमच्या समोर जर गुगल आणि तुम्हाला विश्वाचा अनंत ग्रंथालय तुमच्या मनामध्ये आहे दुसरं, पण तुम्हाला सगळ्या गोष्टीची इन्फॉर्मेशन माहिती लगेच मिळते. फक्त ती ज्ञानामध्ये कशी परावर्तन करता ही ज्या दिवशी तुमच्यामध्ये निर्मिती होईल त्या दिवशी खऱ्या अर्थाने तुमच्याबरोबर मोठे शास्त्रज्ञ निर्माण होईल घडतील असं मला वाटतं. आणि या निमित्ताने तुम्हाला सांगतो की अशी चर्चा करतो या निमित्ताने तुम्हाला वेगवेगळ्या व्याख्याती निमित्तात बोलतात, पण त्याच्यातनं आपल्याला काय घेऊन करत आहे त्याच्यातनं वेगळ्या गोष्टीचा एखादा स्पार्क आपल्या मनात येतो निर्माण झाला पाहिजे. शेजारी त्यातून फार कमी आपल्या मनामध्ये जे आपलं
ज्ञानासाठी आपण म्हणताय की जर भेटला पाहिजे तो त्या दिवशी भेटेल तरी आता तुमच्या आल्यामध्ये शास्त्र निर्माण होतील. या निमित्ताने मी एवढंच होतो आणि तुम्ही मला या ठिकाणी ऐकून घेतलं. या ठिकाणी सध्या आदरणीय भावनांनी मला बोलण्याची संधी दिली. आमच्या महाविद्यालयाला या ठिकाणी चर्चासत्र आयोजित करण्याची संधी दिली त्याबद्दल मी नाशिक सुद्धा या ठिकाणी खूप खूप मनापूर्वक आभार व्यक्त करतो आणि सांगतो जय हिंद जय महाराष्ट्र. काल आपण 6 तास ऐकलं दिवसभर सकाळपासून संध्याकाळपर्यंत आणि संध्याकाळी सोलर कल्चरल प्रोग्राम गव्हर्नमेंटने आयोजित केला होता. सांस्कृतिक कार्यक्रम आपण त्याला म्हणतो. अति सुंदर आणि मन प्रसन्न करणारा होता आणि विजय कॉलेजमध्ये कनिष्ठ महाविद्यालयाचे प्राध्यापक गिरे सर आणि चमूना उत्कृष्ट प्रोग्राम आपल्याला दिला होता आणि मन प्रसन्न केले आणि त्याच्यामध्ये भर घातली प्राचार्यांनी आणि इतकी उत्कृष्ट कमाल सादर केली म्हणून आपल्या दिवसभराचा खटपा लागलेल्या कंपनीसाठी प्राचार्य सरांचे नाशिक सांस्कृतिक मंडळाचे नाशिकच्या वतीने आभार मानतो.
Thank you, sir. Here we come to the concluding part of today's program. In an every way, audience is as important as speaker, and it is essential to express gratitude towards them. Hence, I would like
संस्थेचे संस्थापक अध्यक्ष श्रद्धेय गोविंदादा, मंचावर उपस्थित गेल्या 2 दिवस चर्चासत्राच्या निमित्ताने आदरणीय डॉक्टर गजबीर, डॉक्टर स्वार, बोणापे मॅडम, भारती मॅडम, ITCचे आमचे कॉर्डिनेटर तसेच या वर्कशॉपचे आयोजक मुख्य आयोजक डॉक्टर गवाडे सर, उब्राडे सर, या ठिकाणी या चर्चासत्राला यशस्वी करण्याकरिता सर्व सहकारी प्राध्यापक, विविध महाविद्यालयातून विविध क्षेत्रातून आलेले विद्यार्थी बंधू आणि खूप आनंद वाटत आहे की 2 दिवसीय कार्यशाळा आपल्या सर्वांच्या सहकार्याने आणि आपणही त्या पद्धतीने ते 6 तास बसून या आम्ही केलेल्या आव्हानाच्या द्वारे आपण या कार्यशाळेचा खरंच लाभ घेतला असेल अशी आशा बाळगतो. मित्रहो, जोपर्यंत आपण कोणतीही तकलीफ किंवा मेहनत किंवा जीत लगन ह्या गोष्टी मुलाची बाळगत नाही तोपर्यंत आपण जीवनात यशस्वी होऊ शकत नाही. एक छोटं उदाहरण देतो तुम्हाला. एक मूर्तिकार असतो, तो मूर्ती घडवण्याचं काम करतो. तो मूर्तिकार रस्त्याने निघतो आणि एक चांगली मूर्ती बनवण्याकरिता एक दगड शोधतो. एक दगड त्याला चांगला दिसते तो उचलतो आणि निघतो. पुढे त्याच्यापेक्षा एक थोडासा त्याच्या लेवलचा एक दगड त्याला परत दिसतो. ते दोन्ही दगड दिसतो आणि घरी गेल्यावर तो चांगला दगड त्याला वाटतो त्या दगडातून तो मूर्ती बनवण्याचं काम करतो. हे सर्व मूर्तिकाराचं उदाहरण आहे तुमच्याकरिता. आणि तुम्ही एक दगडाच्या भूमिकेतले आहे की जोपर्यंत तो मूर्तिकार त्या दगडावर आठवड्याचे भाव टाकत नाही तोपर्यंत त्या दगडाला चांगला आकार येत नाही. तर त्याच पद्धतीचे काम या 2 दिवसाच्या चर्चासत्रामध्ये आमच्या मूर्तिकारांनी आपल्यावर केलेलं आहे. मित्रांनो, सांगण्याचं तात्पर्य एवढं की तो मूर्तिकार त्या दगडामध्ये एक चांगला मूर्ती, एक चांगला जीव निर्माण या पद्धतीची मेहनत त्या दगडावर तो आपल्याला कपडे मारून करतो. पण त्या दगडातून आवाज आता बोलतो, आता मला बोलू नाही. तो मूर्तिकार तो दगड बाजूला ठेवून दुसरा दगड घेतो
दुसऱ्या दगडाला अनुभव होऊ की याने नाही म्हटलं आपण असं काही करायचं नाही. तो दगड कितीही त्रास झाला तरी तो सहन करतो आणि तो मूर्तिकार त्या दगडामध्ये एक चांगला मूर्ती तयार करतो. ती मूर्ती देवालयात ठेवण्यात येते आणि त्याची पूजा केल्या जाते. आणि दुसरा दगड होता तो त्या देवळाच्या बाहेर ठेवला. मित्रांनो, ज्यावेळेस लोक हार आणि फूल घेऊन येतात मूर्तीवर चढवतात आणि त्या दगडांनी त्रास सहन केला नाही त्या दगडावर नागर पूर्ण करतात. तर मित्र असं तुमच्यामध्ये दूध आहे. तर जे तुम्हाला या 2 दिवसाच्या चर्चा सांगण्यात आला आहे, तुम्हाला त्रास काढून काढावा लागेल, त्रास होईल, परंतु त्याची प्रचिती तुम्हाला भविष्यामध्ये खूप चांगल्या पद्धतीने. म्हणून मी तुम्हाला एकच आवाहन करतो इथे की आज जे काही या ठिकाणी शिकवण्यात गेलं की तुमचं मर्यादित राहता कामा नये, ते आपल्या अंतर्गत उतरवा. या ठिकाणी जेवढे बसलेले आहेत तुम्हाला सर्व मदत करण्यास तयार आहेत. आपण सुद्धा आमच्या महाविद्यालयाचे संस्थेचे अध्यक्ष भास्कर दादा हे नेहमी तत्पर असतात की आपल्या विद्यार्थ्यांना कुठून काय घेता येईल, काय करता येईल. आम्ही सर्व तत्पर आहोत. आमचे विजयराव कडकडीने बोलतात तुमच्याशी, काम मेहनत करणं काम तुमचं. म्हणून मी आजच्या या महाविद्यालयाच्या 2 दिवसाच्या प्रशासकांना सर्वांना शुभेच्छा देतो. आणि आपल्याला या चर्चासत्रात एवढंच सांगितलं आहे की जब भी आप मुझको पता नहीं मैं कहाँ हूँ, लेकिन इतना जानता हूँ कि कल जहाँ था उससे थोड़ा आगे. धन्यवाद. धन्यवाद सर. चॅनेलवर आलेला पाऊस कृतीसाठी वादन करतो. आणि या 2 दिवसीय वर्कशॉपच्या आभार प्रदर्शनाला सुरुवात करतो. या 2 दिवसीय वर्कशॉपच्या शेवटच्या प्रतिनिधीचे कार्यक्रमाचे अध्यक्ष माननीय रामटेके सर आपल्या महाविद्यालयाचे प्राचार्य यांचे मी मनापासून आभार मानतो की त्यांनी आम्हाला या कार्यक्रम घेण्याची परवानगी दिली आणि आम्हाला वेळोवेळी सपोर्ट केला. त्याबद्दल धन्यवाद. या कार्यक्रमासाठी उपस्थित असलेले प्रमुख पाहुणे गजबीये सर, नाशिकचे अध्यक्ष, यांनी आम्हाला या कार्यक्रमाला जो काही सपोर्ट केला आणि या कार्यक्रमाच्या रूरल एरियासाठी घेण्यासाठी आम्हाला प्रोत्साहन दिलं, त्याबद्दल मी संस्थेच्या वतीने, कॉलेजच्या वतीने सरांचे आभार मानतो. आणि जर असेच कार्यक्रम आम्ही तुमच्या आशीर्वादाने घेत राहू अशी तुम्हाला ग्वाही देतो. या कार्यक्रमासाठी त्यांनी आम्हाला मार्गदर्शन केलं. बारीक प्रत्येक गोष्टीसाठी आम्हाला सूचना केल्या ते डॉक्टर आस्वादकर नाशिक यांचेही मी मनापासून आभार व्यक्त करतो की त्यांनी आम्हाला जर हा प्रोग्राम केला नसता तर आज आपण 2 दिवसाची कार्य आमच्या या कार्यक्रमासाठी उपस्थित असलेले आमच्या संस्थेचे सचिव माननीय डॉक्टर विजयराव टोपे सर यांचेही मी सरांचा नेहमीच सपोर्ट असतो आणि सरांची विद्यार्थ्यांसाठी जी असते ती चळवळ असते असेल की विद्यार्थ्यांनी काहीतरी करावं यासाठी आपल्या कॉलेजमध्ये नेहमीच
हेतू असतो त्या सरांनी आम्हाला सपोर्ट केला त्याबद्दल मी डिपार्टमेंटच्या वतीने सरांचे आभार व्यक्त करतो. तसेच या कार्यक्रमातल्या गॅलरीची प्रोसेस थोडाफार मॅडम आणि बारामती मॅडम उपस्थित राहिल्या. मॅडमने स्पेशली केअर केलं त्याबद्दलही मी डिपार्टमेंटच्या वतीने माझ्या वतीने आभार मानतो. तसेच सर्व स्पीकरचे ज्यांनी आमच्या सरांच्या एका फोनवर सर्व स्पीकर आमच्यासाठी अवेलेबल झाले आणि आम्हाला आमच्या विद्यार्थ्यांसाठी त्यांचं जे काही सर्व वर्षपर्यंत नॉलेज आपण त्या विद्यार्थ्यांना दिलं त्याबद्दल मी सर्व स्पीकर सर ते मला ऐकत नसते तरी त्यांचे मला आभार व्यक्त करतो. माझा मित्र रवी कुटे आणि शैलेश साबळे यांनी माझ्या एका फोनवर मला होकार दिला आणि काही थोडा टेक्निकल प्रॉब्लेम आला पण तरीही ते एक रोल मॉडेल आहे. आणि याच्यासाठी म्हणतो मी की रवी ही आपल्या हातात 45-50 विद्यार्थी आहे पण तो आज डब्ल्यू मध्ये काम करतो. मन करून घेतलं त्यांनी काय ऍक्टिव्हिटी केली आहे आणि असा मुद्दा आहे जो 50-45% होते ज्यावेळेस तो UPSC पास झाला तर त्याला 46% आहे पण तो आज डब्ल्यू मध्ये जिनिव्हा मध्ये काम करतो आणि तो आपल्यासाठी अवेलेबल झाला त्याचेही मी आभार व्यक्त करतो. या कार्यक्रमासाठी ज्यांनी आम्हाला सपोर्ट केला ते आमच्या संस्थेचे अध्यक्ष माननीय भास्कर दादा यांचेही डिपार्टमेंटच्या वतीने आभार व्यक्त करतो. या कार्यक्रमासाठी टीचिंग नॉन टीचिंग प्रत्यक्ष ज्या व्यक्तींनी सपोर्ट केला त्या व्यक्तीचे ऑर्गनायझिंग सेक्रेटरी म्हणून तसे डिपार्टमेंटच्या वतीने सर्वांचे मनापासून आभार मानतो. सरती शेवटी बाहेरच्या कॉलेजमधून आलेले विद्यार्थी असो इथून जाचनपूरचे असो विद्या भारतीचे असो आणि काही फॅकल्टी ज्यांनी या कार्यक्रमाचा येऊन आमच्या कार्यक्रमाची शोभा वाढवली त्या सर्वांची नाव व्यक्त करतो. आणि माझे विद्यार्थी जे आमच्या एका 2 दिवस 6 तास बसले त्यांची चेहरे थोडीफार पडल्यासारखे वाटत होते तर त्यांनी जी शिस्त पाळली त्याबद्दल मी त्यांची मनापासून आभार व्यक्त करतो. धन्यवाद. आणि मुलांना असं विनंती करतो की स्टेजवर यायचं आपल्याला एक फोटो काढायचा बाहेरची पालक पडली नंतर बाहेरचे कॉलेजची पार्टिसिपेंट आम्हाला तुमची गाठ वगैरे या स्टेजवर या मागच्या वर्षी सेक्रेटरी काय या दीक्षित मॅडम चौधरी सर अनिल सर योगेश जंत्र सर ऋषिकेश राऊत सर सोमने मॅडम खुशबू मॅडम देविका आणि लोकेश यांनी स्टेजवर यायचं.